现在如果有人要放 PPT 的话，可以先传到电脑上。
好，
如果要放投影片的话，可以来到电脑前面先放投影片哦。
Hello everyone. Please find your seat. Please about to open. Yeah, because we have some a little bit technical issues, so we will start a little bit. But please find your seat. Yeah. Okay, so thanks for everyone. Good morning for come all the way to this symposium. And my name is Southern Liu, so I'm the main organizer this time, and as well as Dr. Kali. When is here? No. Kali. Oh, another organizer, my colleague, Dr. Wen is here. So during these 2 days, please remember our face. So if you have any question, you can reach us, and we can give you any guidance. And then on the behalf of the Taiwan Coral Reefs Society, I'm very happy to welcome you guys to join this conference and symposium. And this is actually the second time of this international symposium on coral reef in Taiwan. And last time was 2 years ago. However, it's too bad that during that time is a COVID period, so it's not as in terms of the scale, it's not as big as this time. And after the COVID, and now we have the chance to have you guys come here and then establish a relationship and renew the relationship. And I know many of you are not familiar with each other and exchange the information, updated information for research. Before we open up, I would like to have a few people that will have an opening remark for us. And the first person will be the chair of our Center of Ecology and Environment, Dr. Jiang, and his prize support for this time for the symposium. Let's welcome Dr. Jiang.
Good morning. Good morning, Dr. Miller, and Mezaki-san, and Nakano-san, and Colin, and Taidang CEO Zhang先生, and Dr. Tang, Lijijo, and in fact there are many Dr. Lins, Lin Qianxiang, Zihao, and Lin Meifang老师, and Alan Chen, old friends from Okinawa, Nakamura-san, Yamashiro-san, and also Shinsato, Shinkina-san, and Cai Cheng-han 老师, and Chris Joey, and speakers, guests, and friends. Welcome to Donghai University. First, I'd like to sing a song to you all. And with approval from Lijijo Tang Shen-Liu. Okay. Oh, here it is. 白话查甫彼一天，准为你开酒家，只有准的白茫茫，全无朋友来相送。Okay. Thank you. This is probably the most applause I had in my life so far. So let me explain. 白话查甫彼一天, that means on the 15th of August. It's a lunar calendar, so it's a full moon. Last week I realized it's bad for diving, lots of current. 准备离开琉球港, that means the ship is leaving Port Ryukyu. This Ryukyu is not Okinawa. This is a small island off the coast of the southwest Taiwan. lots of turtles. You must visit. So there's just white smoke coming out from the ship, lonely smoke. No friends are here to send me off. Nobody to say goodbye to. Very sad. Okay, this very song was originally sang by Chen Yilang. So Yilang in Japanese is Ichiro. The Chen is like Alan Chen. So I'll picture that. So this beautiful song was written in 1990, 3 years after nearly 40 years of martial law here in Taiwan, when people were strongly discouraged to go to the sea, to enjoy the ocean, because they were spies, criminals, and communists who might harm you or maybe have bad influence on you. So lots of songs related to the ocean were very sad. Accidents, poverties, people waiting at the coast for family members to come, but they never come. Singing songs like that. But after another 40 years of democratic development, we look forward. People in Taiwan embrace ocean culture. People talk about the conservation and sustainability of the coast and the ocean. We care about policies that might influence the ocean. We even had a referendum on Coral Reefs 3 years ago. And we desperately need knowledge-based solutions to the problems and conflicts in marine and coastal environments. And this shows how important this meeting is. And here in Donghai University, we have changed it too. 15 years ago, we hired Alan. And a few years later, we hired Colin. And these two young guys opened a new horizon for research, teaching, and friendship here in Donghai. And I have changed too. I myself is a forest ecologist. I never saw the ocean until I was 18 years old. But last year, I took an intensive diving course at Yudao, managed by Alan Chen. My captain, and my instructor, Sanju, is right here too. So, in the past week, I was with Chris, Joy, and Colin in Penghu Archipelago for an investigation of cryptobenthic reef fishes. And I witnessed the amazing morphological and behavioral plasticity of Tripterygidea. Is that right? Tripterygidea. The triple fins. According to my diving watch, I was underwater for 9 hours and 45 minutes, over 13 dives. This may mean nothing to most of you, but I feel very proud. I will keep on diving and encourage my friends and the family to have license to dive. And with that, I look forward to this exciting meeting. And on behalf of my colleagues here in Donghai, I wish you all enjoy your time here and very productive discussions. Thank you.
Thanks for Dr. Jiang's opening remark. And then the second people I would like to invite is President Dr. Yoshikatsu. Yoshikatsu-san. And thanks Dr. Yoshikatsu for the opening. And then the third person I would like to invite is Mrs. Koshiba. She's now currently the CEO from Palau International Coral Reefs Center. That's welcome. This is.
Hi, good morning, everyone. It's an honor to be here with all the scientists and experts on marine life. And just want to thank, well, sorry I didn't prepare any songs, but for hosting me and my colleague, Dr. Bianchi, to be here to be with everyone who are experts. And the Palau International Coral Reef Center is shifting to one of our main goals is to do coral restoration. And us being here, we want to learn on how corals are being affected by climate change. And hopefully with your expert, you can be able to help us, and with collaboration in the future, that we will be able to learn about how corals can be adapted to climate change and survive in climate change. So I want to thank you all, and it's good to meet everyone, and I'm excited. Hopefully I can meet everyone and all of you. Thank you.
Thank you, Mrs. Pushpa. And the last person I would like to invite to give an opening remark is our President of Taiwan Coral Reefs Society, Dr. Tan. That's welcome.
Okay, good morning. Alan and colleagues, thank you so much to organize this symposium. And Director Zhang, thank you for your great support. And also thank you, special President of the Japanese Coral Reef Society, Makono Kashin. And then the CEO of Palau's Picklets, Mr. Karen Ushban, and also the Taida Jijin Hui, Mr. Zhang. Yeah, thank you. And also invited the whole honorable speaker, especially coming from the distant place, and the dear colleagues and friends and all guests. First of all, again, I want to say thank you to organizer, great organizer and sponsor. Without their efforts and general support, we cannot make this symposium happen. Thank you so much. And 2 years ago, as Alan just mentioned, actually during this challenging posed by COVID-19 pandemic, the Taiwan International Symposium for Coral Reef was organized by the National Taiwan University, actually under the leading by leadership of Dr. Sanhua Yang and Dr. Jiayin Ko. She's now in the Swiss. Along this, and also with colleagues in Afghanistan. And given this stringent international travel restriction and also constraints of the symposium agenda, so we are limited to inviting a single international keynote speaker, who is the Professor Hideyaki Yamashita Sensei. Thank you so much for the time. And then, but today it is a great joy and sense of the achievement. We are welcoming multiple international scholars and participants to join us in the person. And it's reality encouraging for both organizer and also the society here. And actually, the primitive objects of this symposium is to foster regional cooperation and enhance educational and outreach activities related to coral reef research. And then, in the light of profound global environmental change, like say the climate change, all this, we are seeing the many coral research questions actually cross the national borders. So, so underscoring the urgent need for international collaboration. For instance, last year I attended the Japanese Coral Reef Society annual meeting. And then I saw detailed observation and surveys conducted in the Okinawa, Yaeyama Islands. And then, but this area actually mainly located from located merely 200 kilometers away from Taiwan. And however, beyond the Japanese border, there's a notable absence of all this crucial data in Taiwan. While some Taiwanese scholars may possess relevant data, but lack of mutual cooperation mechanism and integration has hindered our ability to leverage comprehensive data to tackle the broader scientific questions and provide more complete evidence. So similar gaps in data sharing and collaborations exist between the Philippines and Taiwan. So next time we will try to invite more scholars from the Philippines. And then, so therefore, I passionately advocate for the collaborative initiative involving Japan, Taiwan, the Philippines, Palau, other countries to establish this transnational crucial research area. I like to call it a cool research zone. Okay. So this initiative aims to promote extensive international academic research, exchange on coral reefs, enables us to address more significant, concrete, and expensive scientific issues and questions. So this symposium is committed to serving one of exchange platform to facilitate this academic and educational cooperation. So finally, I hope that each of you will not only gain the new knowledge from this symposium, but also opportunity to engage in more discussion and forge new connections and exchange ideas. And I wish the symposium a smooth and successful conclusion and pave the way to the future advancement in the coral reef research. Thank you so much.
Thanks for Dr. Chan's our president for the opening remark. And then before next one will be our keynote, but before that, I also would like to add a few notes. We also like to thank the funding source from our three different government branches. The first one is the National Science and Technology Council, and the second is Ocean Conservation Administration, Ocean Affairs Council, and the third one is the National Academy of Marine Research. Without their funding support, it's not going to make this symposium happen. So thanks for their support. Thank you. And then please sit tight. And then now we have to have a group photo first. So maybe you sit here, and then we have a cameraman here, and then please just mark out and then take a photo first. So please probably move a little bit to the center, yes. Sorry about that.
I was assigned by Solomon here to introduce the first apprentice speakers, Professor David Miller Chen, Dr. David Miller, not Chen. Chen is his student. It's my great pleasure to introduce my supervisor, my PhD supervisors here. And I would also want to thank Dr. Tan give this honor time to Professor Miller to come here to share all his life experience on the coral genetics, molecular, and now genomics. And I just prepared 27 pages of introduction to this study, but he just told me keep it short. Okay, I will keep it short in about 2 minutes. I give you all information about the first of Dave. Dave is the first researcher to clone the gene from coral back to late 1980s. Okay, and Dave is the first lab to home the Symposium algae genes in the world. Dave he has the lab and all the lab members to conduct the very first molecular phylogeny of coral evolutions. Dave had the lab to sequence the very first genome of the coral. It's Acropora glabra. And till now, still the best genome in assembly and also annotates. And now Dave is the leading scientist in the world to keep going on to unlock the mystery of the symbiosis between coral and their symbiote, including coral and including algae, virus, and so on and so on. So, and through the time, I mean, the last 40 years, he's been training over 42 PhD students, including me, including Chuya, including Mayvon in this group. Okay. So, without further ado, let's welcome Dave to give his talk. Dave?
Forgive me, technical problems, absolutely. First, I want to say a couple of things on the bio at the outset. I thought that was an amazing introductory speech from Professor Chung. I hope my pronunciation is something like correct. You know, I have no idea of the difficult past Taiwan has lived through. So that was great. But I hope I don't have to try and follow you in terms of singing the song. It's a bit beyond it. You don't want to hear it. Put a nail in the coffin of the Caribbean Reefs. Caribbean Reefs suffered terribly from all manner of damage, but I think coral disease is really not looking here. Okay, so as I arrived at JCU, we made some progress towards isolating DNA, cloning sequences, cloning genes, and so on. The progress was really quite slow, gene by gene, until this paper in 2011 from Norisato's lab, the lead author being Choya, observer being Choya Sensato. And this really, the publication of the first complete genome sequence for a coral really enabled a whole new domain that really changed coral biology, I think, enormously. One of the things it enabled was some comparative genomics. Comparative genomics is quite, it's quite a respectable literature on comparative genomics. In this case, what we could do was compare the genome of Acropora with that of the sea anemone, which had been published slightly earlier. Sea anemones, essentially corals without skeletons. So the hope was that by comparing these two genomes, we might get a handle on what new genes, what new things happen in the genome to enable things, coral-specific traits like calcification and symbiosis.
bacterial interactions. Lots of work has been done on the bacteria associated with corals. There's an enormous literature on describing the microbes associated with a diverse range of corals under a diverse range of conditions from a diverse range of places. But really, I find a lot of this literature pretty woolly and hard to generalise about, hard to interpret. There's been some experiments on exposing corals to whole bacteria, usually at totally unrealistic concentration, or hitting corals with bacterial traps, lysates of suspected pathogens, and so on. But much coral immunity has simply been based on identifying coral homologs of mammalian-specific mammalian genes known to be involved in innate immunity. It's correlative work. Even RNA-seq experiments, it's all correlative work. We think these genes are involved because their expression changes under certain conditions. And I think there's far too much of this kind of correlative work, far too little real experimental work being done on corals to date. Okay, so we played this game, Troy played this game too, comparative genomics between sea anemone and coral showed that in the early days, even at the outset of this paper, this figure is from the original genome paper. What you can see are the primary extracellular pattern recognition proteins. That's the toll-like receptors and the interleukin-1-like receptors. The repertoire that was found in the early days from Acropora is here. And you can see it's much more complicated than the limited number of similar receptors seen in CNN. Hydra, as it's typically Hydra, represents a really derived state. So it's clear that Acropora has a much more sophisticated extracellular pattern recognition system. And the same is true of the intracellular pattern recognition proteins. The NMRs, the NAP domain containing proteins. Acropora has 500 or more of these kinds of proteins, whereas the sea anemone and MAM have an order of magnitude fewer of these. So the ability of corals to recognize a wider variety of things and discriminate between a wider variety of things, which is implied by the presence of things, suggests an improved ability to resolve potential pathogens from potential symptoms. All right, so coral diseases, as many of you know, affect many corals, and their names, the names of these pathologies, really just are descriptions of the macromorphology involved. So most coral diseases are named based on what you can see. There's white band, yellow spot, stony coral tissue loss disease, etc. But this doesn't tell you anything about the causative agent. Very few bacteria ever met Cox postulates. You can't isolate them and use them to reinfect corals. It looked like Vibrio coraliolyticus might fit that bill, but that word can't be reproduced. Probably because the pathology was that the pathogenicity genes may have been lost. So it looks to me increasingly like most of these coral pathologies really are simply normal components of the bacterial microbiome that have got out of hand. There are many facultative pathogens associated with corals. You can find them in healthy corals, but at much lower densities than in diseased corals. So that's kind of the hypothesis that we work to. And amongst those, for example, white syndrome infection on Acropora. White syndrome if you isolate bacteria from around these kinds of regions, you can see lots of Vibrio species that are normal components of the microbiome of healthy corals. All right, so I think that I'm increasingly thinking that corals are, well, it's clear that corals are more susceptible to disease when under stress, and the disease in corals is predominantly driven by facultative pathogens escaping regulatory controls. Okay, so forgive the cartoon. I hope some of you may have seen it before, but in thinking about coral microbe interactions, I think it's useful to think about the mucus layer as a sort of war zone in which conflict occurs, partially to protect the coral from invaders from outside. So microbes are present, a wide variety of microbes are present at low density in seawater. The concentration is greater in coral mucus because bacteria are trying to use, many bacteria want to use coral mucus as a source of carbon energy. So higher density of bacteria, probably lower diversity, but these bacteria are slugging it out in the mucus layer because they're competing for resources. And they compete with each other using antimicrobial peptides and most other things. Antimicrobial peptides are the kind of go-to first line of defense in a wide variety of animals. And so eventually we decided to try and characterize some of these. I should have said back there that some microbes actually make it into the coral tissues predominantly, but not exclusively, endozoic. And we'll return to endozoic, I hope. So, antimicrobial peptides. So since pretty much all animals make antimicrobial peptides too, we set out to look for some. It surprised me that only a few years back, only one AMP had ever been identified in corals. So we set out to find some more. And after much toing and froing and some help from Choya, we came up with this as the best candidate amongst a long list. This protein has the characteristics, many of the characteristics that are associated with the antimicrobial peptides. They're quite tricky to identify because there's low sequence similarity, but there is three-dimensional protein structure similarity amongst limited structural conservation. And so this protein, long protein leader sequence, which is chopped off during secretion, there's a pro-protein sequence which is also removed, leaving a relatively short peptide which has lots of basic amino acid residues, and it's also cysteine rich, both of which are characteristics of antimicrobial peptides. So we had some of this made chemically. That's one structural similarity with both dabicornin, the first AMP clone from corals, limited sequence similarity, but structural similarity also with a diverse range of sea anemone toxins. Had some of the protein synthesized because it's quite short, and my friend Renee Augustin demonstrated activity against a wide range of bacteria, both medically and ecologically relevant bacteria. And we published this, studied this protein 2 or 3 years back, so I don't intend to dwell on it, except to say that the protein is first really highly upregulated in the planular larva, especially during the pre-settlement and early settlement period, but it's also expressed through adulthood. All right, so, that's the classical kind of sign of immunology, immunity in corals. Coral microbe interactions are to some extent controlled by antimicrobial peptides. But what I'm going to show you next is, I think, much more novel, much more interesting. It is very novel and interesting. It's also very topical because I think the paper was slated to come out either today or tomorrow in Current Biology. After an amazing slog, this paper should be out any day now. And what we describe is a novel kind of mechanism that we think corals employ to control their microbiomes. Alright, so before talking about what we did, I need to ensure that you're kind of a little bit up to speed with quorum signaling. I know some of you know a lot more about this than I do, but quorum signaling is how bacteria communicate with each other. They communicate with each other using small molecules, and they do so that they know, primarily, so that they know how abundant the same kind of bacteria is out there. What's the density? How many of you are out there? That's communicated using small molecules, and acyl homoserine lactones are, I think, the most common, certainly the best characterized, probably the most widespread in terms of use. Family molecules used by bacteria to communicate with each other. AHLs, acyl homoserine lactones, they all have this homoserine lactone head, but the side chain, the acyl side chain, varies a lot in terms of length, number of carbon atoms, and also degree of substitution. So AHLs are the most widely used quorum signaling molecules in gram-negatives and gram-positives. I forgot to tell you that the quorum signaling is used oftentimes to coordinate gene expression, so that bacteria turn on genes when they reach a certain density. They're able to turn on genes for example, pathogenicity factors or biofilm formation or whatever. So, acyl homoserine lactones common across gram-negatives, but also in some gram-positive bacteria. And there's an additional layer of complication here in that bacteria communicate using AHLs, but many bacteria actually compete with each other by making enzymes that cleave or inactivate the quorum signaling molecules of their competitors. And this is called quorum quenching. Quorum quenching occurs in bacteria, three mechanisms predominantly. That is cleavage of the lactone ring, lopping off the side chain by acylases, or modification of the side chain by oxidoreductases. So, it's known that in addition to antimicrobial peptides being thrown at each other by microbes, there are also quorum signaling and quorum quenching activities present in coral mucus. There's a literature on this. Presumably these are derived from bacteria. Alright, enter Yvonne Weiss. Yvonne, former PhD student of mine, very bright girl, knew a lot more immunology than I did. And what she did was a neat kind of experiment that I thought would fail miserably and cost me a lot of money. She decided to challenge adult corals with a defined immunogen and look at what happened in the first hour of exposure to, in this case, uramyl dipeptide. This is a bacterial cell wall component. It evokes a very well understood immune response in mammals over a short time window. She wanted to see what would happen within 1 hour if you expose corals to a challenge using this. And to my great surprise, we found quite a few genes up and down regulated. This was our first RNA-seq experiment. It was based on very short weeks and not an enormous number of them. It cost me an enormous amount of money, but actually it yielded some really interesting results. Genes in green are upregulated, genes in red downregulated, and the ones highlighted in yellow are the ones we wrote the paper. So the ones in red are actually genes that we'd seen before in other contexts. Some of them made sense, made perfect sense in terms of what we thought might happen, what happens in other things. But some of them I really didn't want to think too much about at the time, such as the one indicated with the red arrow, Coral glycine hydrolase. So why didn't I want to think about it? Well, database searching, the best matches were bacterial, and I thought, well, could be contamination. Let's not worry about that. Let's write the paper based on something else. And that is the paper we wrote based on something else. You can see my penchant for very long and complicated titles. Coral glycine hydrolase. Right. This is a coral sequence which encodes what's called an N-terminal nucleophile hydrolase family member. Sorry, it's a big mouthful. Abbreviated to NTNH for points. These proteins are generally secreted. Removal of the secretion signal leaves an N-terminal cystine residue that's involved in the catalysis. So this is where the name comes from. The N-terminus is a nucleophile involved in the chemical reaction, biochemical reaction. The coral protein has a C-terminal ER retention signal, but it's not a perfect ER retention signal. We'll get to that shortly. The complication at the time was that there were no close homologs in benzones. All the close homologs were from bacteria. Complication. Right. So this is the sequence. A couple of things I should show you in connection with what I just said is the leader sequence, which is locked off during secretion, leaving this cysteine residue at the N-terminus. As part of can then form the active form. The residues in green are the active site residues. The shaded regions are binding loops. So in addition to the C-terminus, the N-terminus has got this KEL sequence. That's not quite a perfect endoplasmic reticulum retention signal, but it goes to the endoplasmic reticulum secretion signal. And it's tempted to stay in the ER, but it leaks out slowly, but at least that's what we think happened. A perfect ER retention signal is KDEL. This is a partial retention sequence, and we think what happens is it leaks from the ER slowly into the mucus layer, where it's most likely active. All right. So here's a simple bar chart based on all the data we had at the time, showing that developmental expression of the protein. You can see it's strongly upregulated during the settlement phases, but thereafter downregulated until you hit it with a new challenge. So Yvonne did this sort of developmental comparison just because we had the libraries, we had the information with which to ask the question, not because she expected it to be developmentally expressed, but it is, and it's most heavily expressed in the planula to larva, planula to polyp transition. Okay, and we had actually seen this gene before. It was previously known, it had turned up as a large inner line screen for genes that are expressed in that group, that is, during the settlement metamorphosis period. So what's common about settlement and immune challenges is settlement itself is a kind of immune challenge. Hopefully this will work. Yeah, but in this video by Chris Brunner, you can see coral larvae undergoing settlement and metamorphosis on the surface. they're probably attracted to that surface by specific bacteria. But that surface is also going to be covered with potentially hostile bacteria. So it kind of makes sense to upregulate your immune system during that period. But that's the uniting factor about immune challenge balance and the settlement process, is that you're going to face immune challenges in both situations. So far, so good. And then, I had one day I was reading, and reading stuff really quite peripheral to anything to do. And I had one of those moments which I would call penny dropping, or John Mattock has called these epiphanies, where you suddenly see something quite clearly, or you suddenly think you see something quite clearly. So that's happened to me maybe twice in my life, and this was Saturday. So I was reading about these proteins, bacterial proteins, which belong to the same protein family as our coral protein. And their primary activities are totally different, but both of these can act as hydrolyzers, can act as pore quenching activities against AHLs. The primary activity is something else. They've been cloned as proteins that carry out different functions, but they also belong to the same protein family as our coral protein and have the distinction of cleaving AHLs. So there's your hypothesis. Bingo. Maybe the coral protein is involved in core quenching of these signaling molecules, AHLs, used by bacteria associated. So we set out to test that. In fact, the activity that we think we have is likely to be an acyl-A is likely to do this, lock off the side chain of AHLs. This process has only been seen in bacteria previously, but we suspected that the coral protein was doing this. In other words, we suspected core quenching was going on by a coral protein. Alright, so to establish the activity of the protein, to test its ability to do what we think it does, we started to make the protein it was easy to express, it was an absolute nightmare to purify. It's largely insoluble, solubilizing it activates a large amount of protein, etc. But eventually we got enough of the protein to test the activity. The activity assays are also a bit of a nightmare. I'm going to just briefly try and describe the assays before showing you all of the results. So with C6AHL, that's a 6-carbon chain, side chain AHL. This is a reporter system where if you add the AHL, you get light output over the next few hours. So light output is shown here, the solid line. If you add the coral quenching activity, the coral protein, you get a strong reduction of light reduction, corresponding to removal of the AHL. So that's what the dashed line is. The dotted line is another control, which is heat inactivated coral protein. So you can see, I hope, that the coral protein is active against the 6-carbon chain AHL. It's also active against other short chain AHLs, C6, C7s, and 8s and 9, to varying degrees. Got to say that you can't do anything sophisticated with this assay. You can't even compare activity across different AHLs because the system responds differently. Light output differs with different AHLs, and you can't do kinetics or anything sophisticated. But it does show that the coral protein does have activity against some AHLs. Alright, so to cut a long story short, there's not just one of these genes in the coral genome, in the appropriate genome, there's actually 6, which generate more than 6 proteins through alternate splicing. The gene that we first cloned is this one, but it has a sister gene just upstream, and there are 4 other genes likewise organized as linked pairs, but the pairs are unlinked. These are similar in sequence, they're recognizably similar in sequence, but they are sufficiently different to be clearly distinguishable. And that's the pattern of organization across the cropper. So the same organization in Cropper Butifera, and also in this, which is a member of the Cropper attenuous clade. The Cropper attenuous clade is basal within the Cropper taxon. So that's how it is in the Cropper. It's quite different in Multipora, and different again in other corals. But these genes are present in a wide range of corals, I should say. Alright, so the first gene that we cloned, I've shown you this before, is down-regulated in adults, it's up-regulated in immune challenge. And you can see that here, sorry, a more sophisticated kind of expression profile. Up-regulated is red, down-regulated is blue, purpley coloured. This is over, through early development, through early secondary period, and here's where we use the second inducer. This is the first gene we cloned, that's its sister gene, it's expressed in the same pattern but more weakly. But the other genes are not expressed significantly through the early period. But we know that they're expressed in adults. So we'd like to think that these other variants, this is a member of, these are members of the 2 other gene pairs. They're expressed predominantly in adults. We think they're probably involved in modifying the adult microbiome. Okay, so we've got this kind of coral quenching activity from coral. It's a first in any animal. Almost at the same time, Sebastian Frauner in Thomas Bosch's lab found an activity in hydra which is also able to coral quench by a totally different mechanism. It's an oxidoreductase that modifies the side chain of some AHLs. But unfortunately, this hasn't been cloned. There's no sequence data. So we don't know what it's related to, and we can't look for a coral hormone. Is this a Cnidarian-specific phenomenon? I think we've just found the tip of the iceberg, and that this kind of thing happens in other animals. And there's some kind of evidence that it does happen even in mammals by, again, a different mechanism, by lactinase activity. But these papers are a bit controversial, I have to say. All right, so does the expression of the protein correlate with changes in the microbiome? Well, we know the microbiome changes radically through this settlement period. The arrowhead is use of the settlement cube. Microbiome changes. These are just shown at high level from in the pie charts here. So the microbiome changes. So yes, the microbiome changes, but we know a lot of other proteins with known or suspected AMP antimicrobial activities are also expressed at that time, including the AMP that I showed you, but also proteins like apextrin, which is a MAC-perforin domain protein likely to also antibacterial. So there's no kind of direct connection that we can demonstrate between changes in the microbiome and expression of this coral quenching activity, but we think it's part of the arsenal, part of the weaponry used by corals to manipulate the associated microbes. One final thing, if I've got time. So it's a bacterial mechanism hijacked by corals. The only close homologs of the protein are from bacteria, uniquely from bacteria. but we don't think this is certainly, it's probably a natural gene transfer, but it's a very old one. Why is it a very old one? Well, I'm sorry, I can't speak the detail, I'm sure. But these are coral proteins. We recently found some related proteins in hydrozones. But in molecular phylogenetics, the clade containing both hydrozone and anthozone sequences is monophyletic and well resolved from all of the bacteria in pink or red. So, if it's a lateral gene transfer with a single origin, it happened before hydrozones and anthozones diverged, which is a very, very long time ago. It's early Cambrian or even earlier, possibly. We know Endozoicomonas can make it into the coral tissue. It turns out that thanks to sending tanks, say Endozoicomonas doesn't use AHL-based coral signaling, so it escapes this kind of surveillance, this kind of defense mechanism. But on the other hand, vibrios, many of which increase in abundance in pathologies, vibrio species rely on AHL-based signaling, and we think that they are the primary targets of the coral poaching activity. So, Endozoicomonas, beneficial symbiotes, don't use AHLs, but vibrios, facultative pathogens do. And with that, I just want to thank again Ben Mason, who came as an NSF postdoc to work on opsins in corals, did that, but also started both of these projects. Amazing guy. Alana, who has been instrumental in the protein expression of Bruno and Sebastian and Thomas's lab, sending Tang, Academia Sinica. The OIST Marine Centre staff, particularly Duo and Mary Collins, who's former Dean of Science and Dean of Research at OIST, who was very kind to us. And again, we are funded by the ARC and OIST. And with that, I hope I haven't gone over time. Thank you very much. I hope that wasn't too long.
Okay, thanks Dave for such an interesting excellent talk. Now we still got 5 minutes, I think. So we're open for questions for Dave. Any questions?
Just dying to get to coffee.
Yeah, that's any.
Thank you, Dave. It's a wonderful talk, and also very interesting discovery. My question is very simple. It's about this quantum QQ's protein. Have you measured, except only life stage, but also is it the abundance of this protein also related to the certain environmental change, for example, heat up or something, nutrition, something like this?
Would love to do that, yeah. Sorry, Stanley, great question. Yeah, would love to do that. I haven't quite got there yet. I mean, my idea is to try and connect these activities with coral health in the environment. For example, how heat stable is protein. What happens to them if you take them to 35 degrees? You change the structure drastically, you inactivate them, or what? So, yeah, we'll get there. But this was just really the first step, is to try and characterize activity, demonstrate activity, make sure people are aware. You should talk some more about what it would be interesting to do next. Thank you.
Any more questions?
As one, I'm like I don't really know much about the bacteria, but because you talked about the immune, is there a way we can help them boost up their immune system so they can be more against those infections?
To crank up their immune. Yeah, well, this one goes. This one's louder. Sorry, I mumbled. Yeah, again, would love to know that. I mean, it's clear that coral health is compromised if you heat stress corals. For example, in heat stress, corals are more susceptible to disease from fragmented pathogens. What's the mechanism there? If you could engineer a more heat-stable version of the protein, perhaps that might help. In theory, some sort of sophisticated gene modifications could help, but we're a long way from that technology. First, we have to understand what sort of changes are needed, but second, the methodology for transformation of corals, for genetic transformation of corals, is really not there yet. There's no established reproducible technique, despite what you may have read about GMPs and from bats. That's nowhere near reproducible, and it's nowhere near reproducible kind of approach. So, love to know the answer. Great question again. Come back in 10 years' time, when there may be no coral reefs, I guess.
Any questions? I have two questions. The first one is there's a slide show the different expressions in different stages of the coral larvae, with the H1 gene and 2 copy. And it's quite obvious that there's one copy of the gene expressed quite strongly, but after the prenatal stage. Yeah, but the other one is less. So is any kind of specifics reason why it's these are different expressions between the two copies of the gene? At least with this one.
Yeah, I'm sorry. I probably should have shown this. This is based on a really neat data visualization tool that Ira Cook developed, which will be public very soon, I hope, where you can look at tightly staged expression of a whole range of genes. This is a beta test version, and I don't think there's really, it wouldn't be, it's not 100% reliable, I've got to say. And I don't think there's really a significant difference between the second one. You're talking about expression. Yeah. I don't think there's really a significant difference in actual expression. The visualization system is easy to implement, but the algorithm underlying it is quite complicated. And I wouldn't rely on it to make very fine distinctions. So I probably shouldn't have shown it.
The second question is, through your talk, I will actually keep thinking that the gene should be horizontal transfer from the tree, but until the tree shows, it's quite obvious that they might occur way before the early evolution of the Nigerians as well. So this reminds me that the early work that you published, that the similarity between Nigerian and actually the later Megazoan, the advanced Megazoan, and compared to C. elegans or Drosophila.
Yeah.
Would you be able to comment that why you keep finding this, and also the setting of the whole genome, it looks like a really ancient body?
Yeah. I think I get the question. Yeah, I mean, I want to say that the paper you're talking about was the current biology paper in 2003. Yeah. That was my first epiphany moment, and this is my second epiphany moment. So it's kind of classic. All right, so why do we think this is a lateral gene transfer? Well, maybe it's not. Maybe it's a gene that was there a long time ago and has been lost from other animals. At the very least, it's a very old lateral gene transfer because there are related genes. We first found related genes in robust corals as well as complex corals, and well, that division happened 450 years ago. But finding the database has been added to substantially in the last couple of years, and we found some clearly related sequences in hydrozones. And that division is ediacaran, probably. So if it isn't an ancestral gene, it's always been there. It's a lateral gene transfer that occurred prior to that hydrozoa amphizoa division. So, well, yeah, you decide.
Okay, I think it's about time. Okay, let's just thank Dave again for his wonderful talk.
So we will have a 18 minutes break. So it's the time for the presenter to switch to the pair section. And we will have a tea and drink upstairs. Yeah, the dessert will be in the afternoon. Yeah, but now you guys can switch section. Thank you.
有需要上传海报报告的 PPT 的话，可以麻烦帮我来前面上传一下吗？谢谢。
this session, we have two speakers. One is Dr. Joseph Lee Bastista. Yes, I'm not sure I have pronounced well. And also me, Mei-Fang Lin, and we will give the talk to share our research. So let's welcome our first speaker, Joy, please.
Morning, everyone. Volume's okay? Yeah. My name is Joey, and I am based in Australia at Griffith University. And today I thought I'd talk to you a bit about some of the newer genetic tools that we're actually using to not only track biodiversity on coral reefs, but also as a means to benchmark the recovery of coral reefs where restoration initiatives have been put in place. I've moved around a lot of institutions, a lot of postdocs and jobs. Probably where I got the best introduction to some of the genetic tools that I'll talk about today was in the Trace and Environmental DNA Lab, the Trend Lab at Curtin University in Perth, Western Australia. And here, the picture on the top, you can see that they built these modules that are contamination-free, that are designed for the purpose of isolating trace and degraded sources of DNA. When I entered this large lab group, they were focused really on looking at protein samples, so ice cores, to look at the DNA that they could amplify from those sources. Dust and bones, so archaeological sites, they would grind up a lot of the small, unidentifiable bones to figure out what lived at a particular place at a particular point in time. And a lot of fecal samples to study the diet of large mammals in this area. But given my background's marine, we had lots of chats about how we could convert those tools to use in our oceans. And so we started to work on the isolation of environmental DNA in seawater, otherwise known as eDNA or EDNA, however you want to pronounce it. But essentially it's just genetic material that you get directly from environmental samples. And the nice thing about it is it's largely non-invasive. It's non-destructive. In our case, we're literally just scooping a bit of seawater, filtering that seawater, and then capturing all the DNA that we have in that particular sample. For those of you not familiar with the process in the lab, I'll keep it very basic, but essentially you isolate DNA from whatever source that you might be sampling, whether that's a water sample or a sand sample or a soil sample. And then you're using barcodes to identify particular species that may have shed their DNA into that sample. So in this case, we're using polymerase chain reaction, or PCR. In this case, it's a special PCR, so we're adding tags onto our products so that we can combine all of the different PCR products into a single next-generation sequencing run. So we now have the ability to sequence millions and millions of reads in a single run. And then using bioinformatic tools with those tags that we've applied to samples, we can separate them out, and then we can start to match them against public genetic databases. The most popular one that you've probably heard of is NCBI GenBank. And we also can try to match our unknown DNA sequences to custom databases that we generate ourselves. The nice thing is that you don't have to have those fancy labs set up, and you don't have to do all the work yourself anymore. There's lots of commercial labs available in Australia and New Zealand. There's also lots in Europe and in America, where you can literally send your sample that you've collected in your field trials to these labs, and they do all the work. And they're often quite responsive and very good to the needs of your experiment. So these are examples from Australia and New Zealand. Some of these companies will do species-specific tests. So let's say, for example, in the top right, you're interested in the platypus, a very Australian animal. They'll design an assay where you take a sample and it will identify if platypus DNA is there or not. Some of the other labs have broader assays, so they're looking at lots of different animals in a particular sample. And they say, but the collection of the sample would essentially be the same. What do we know about this field of eDNA? Well, we know it's becoming quite popular. It's a fairly new field. If you look at this schematic here, some of the terms were only really coined about 10 to 15 years ago. And you can see that the number of publications through time has really started to increase exponentially. In this graph, ends at 2017. If you extended that to 2024, you'd see it kind of go off the charts. And you can see that there's lots of different types of substrates that people use. They'll focus on water samples or maybe surfaces that they swab. Now people are getting into filtering the air, so they might stick a filter in this room and pick up all of the DNA that's free floating around, and then amplify that DNA to identify what's in the area. It's kind of past its growing phases and bumps and bruises, and so we know that it works, and we know that it works quite well. So lots of labs have now started to apply global studies. So the top example here is looking at plankton toes across our tropical oceans. There's lots of groups that are working on some of the technical aspects, whether that's bioinformatic pipelines or some of the lab aspects of amplifying bits of DNA. There's lots of groups that are using it for biodiversity surveys, and I'm going to talk a lot about that today. And then finally, some groups are using it to detect much larger things like apex predators, so things like sharks. And there's also groups that are using it to detect the presence of whales in our oceans. Here's just one example of some of the work we've done in terms of biodiversity survey. And this is kind of in the more early days of developing this approach. And what we wanted to do here is really take a tree of life approach to eDNA, in that we didn't do a single genetic test. We were actually using 10 different primer sets to amplify every single plant and animal that might have shed DNA in our particular sample. And you can see that some of these tests will pick up a whole bunch of different taxa, a whole bunch of different eukaryotes, and then some of the tests will only amplify the DNA from fish or mammals or crustaceans, for example, that's in your sample. And what we found in this particular study, we did just a single seawater sample. So we had 9 liters of water from a coral reef, the southern end of Ingloo Reef in Western Australia, Coral Bay, where we applied all these different metabar coding primer sets or assays that targeted 5 different genes, generated millions of sequence reads. But the big picture is that we picked up a lot of different phyla of organisms, many from chordata, crustacea. But what this really tells us is we can now start to look at an ecosystem-based management approach with eDNA as opposed to focusing on a single representative indicator species. So we get a lot more information that we can apply more broadly. Another thing to note about eDNA is, like any survey tool, it is biased. So if you went out and you did a transect tape, you're obviously missing a fraction of the biodiversity that's there, and eDNA is no different. But in this study, we wanted to kind of figure out where those biases were. So what we did is we compared water sampling for eDNA with BRUVs, which are baited remote underwater videos, so video cameras under the water that are capturing all the fish communities that are there. And we put this out in seagrass and rocky reef habitats. And this is kind of the big picture data that we got out of it. And on the bottom right is a picture of what a BRUV is. So essentially, there's a bait bag in front, and it records video over a set amount of time, and we identify all the fish that are in that frame. So what we found is that the two methods were very good at separating the community composition of fish from the seagrass habitats versus the rocky reef habitats. So you can see the separation between the similar symbols. But then there's this great divide in that the BRUVs were picking up a different fraction of fish in the water column than the eDNA was. So the BRUVs tended to pick up a lot more of the large predatory fishes that are attracted to the bait, that are easy to identify on a camera. And then the eDNA was actually detecting a lot of these small cryptic or schooling fishes, which often have much more biomass than some of the larger fish that you see on a reef. So they're actually complementary approaches. We've also started to apply this technique on tropical reefs to look at human impacts. So in this particular study, we worked with Reimer Sensei out at University of Ryukyu, and we went to Okinawa, and we sampled eDNA at a number of sites that had different levels of human impact. So these numbers here in the circles represent an anthropogenic impact scale. So really low numbers were really marginal habitat, very heavy human influence with sewage outflows and lots of different use, whereas the high numbers represented more isolated, pristine habitat. And what we did is we then compared some of these eDNA data sets amongst the different impacted sites. And if we look at a general gene, so it detects a lot of different metazoans, a universal primer, we find very good separation between different sites in terms of the species composition, how it changes with impact. And that was kind of like a pilot trial to figure out if it does work and how it works. And so going forward, I think it can be a very important tool to identify tipping points in particular environments and some of the impacts. So the two little plots on the left here are basically a replicate of the last slide. But if you notice panel C, the thing that we started to notice is that some things may disappear entirely with human impact. So a lot of the highly impacted sites lost most of the coral cover at those particular sites, or we didn't pick up the presence of hydrozoans. But the more interesting thing is that some organisms don't get eliminated entirely, but actually change the types of species that are there. So for sponges, for example, we didn't lose sponges at the low impacted sites, but the sponges that we were picking up were a whole different set of species than we actually saw at the sites that had much less of impact. So I think this can be an important tool for benchmarking or providing snapshots very quickly of particular sites, and then as a means to track the progress of restoration efforts that you might put in place at these impacted sites. How else can we support eDNA? There are some gaps that we have in this tool. One of the big ones is a lot of the local reference databases. So DNA barcodes for a lot of species around the planet are just not there. We don't have them. So to bolster that, I had the unique opportunity of being curator of fishes at the Australian Museum for the last 5.5 years before I moved to Griffith. And so there was lots of undescribed biodiversity just sitting in jars and shelves. What a lot of people don't know is a lot of those fishes in jars are actually reference data that get incorporated into publicly accessible biodiversity databases available online. One of the big ones is Atlas of Living Australia. So it represents a particular fish species that was sampled at a particular location at a particular point in time, right? And a lot of the material that we have at the museum is great to then generate DNA reference libraries to support the environmental DNA work. And why is that? Well, because they've been expertly identified by ichthyologists, and they have a known data location where they were sampled. So we have a physical specimen to back up the DNA barcode. So when I was based at the museum, I kind of made a start at generating DNA barcode libraries for lots of the fishes that we had in our collection. A big bulk of the fishes that we actually have at the Australian Museum were sourced from Lizard Island. A lot of people don't know that Lizard Island Research Station is owned and operated by the Australian Museum. It's over 50 years old now. It's one of the landmark remote research stations in the world. And they have a very good knowledge of the fish species that are known from Lizard Island amongst a lot of other marine organisms. And so we kind of made a start on a barcoding library. But then at a certain point, people with a lot more drive and a bigger vision and a lot more funding kind of surpassed us. And so this is CSIRO, also based in Australia, started this National Biodiversity DNA Library Initiative. And so we've now started to feed a lot of our tissues from the museum into this. So their aim is to barcode every single animal that exists in Australia over the next 5 years. And they started off with the fishes because it's one of the most taxonomically resolved groups. And they have a nifty way of doing that through genome skimming, so they're not generating just a CO1 barcode for every tissue that we provide them with, but every barcode that you can imagine that might be useful for any future project. So it's a great initiative that they're working on. So one of the things we wanted to do is we wanted to look at whether this approach where you're isolating DNA from all of the animals in your water sample, whether it's robust to future monitoring applications. So apologies that this is not a tropical reef. This is a temperate estuary, Sydney Harbour. So this is some of the work that we did about 2 years ago, where we went around monthly sampling in Sydney Harbour to use these little self-contained eDNA kits that we then applied a number of genetic tests to isolate all of the fish, birds, crustaceans, mollusks, plants, algae, and so on, that have shed their DNA into the water sample. And they're really great because they're intended for general use. So we actually had a lot of everyday citizens helping us out with the sampling when we couldn't get out to a particular location. And this is just an example of one of the sites where we sampled. So this is about 12 months of sampling in a particular location in Sydney Harbour known as Parsley Bay. So you can see we picked up a lot of different fish species and worms and crustaceans and things like kelp and sponges and seagrass. So it kind of gives you a broad picture of what's going on at a particular site. And for those of you really interested in fish, we picked up a lot of the larger iconic fishes or recreationally important fishes. For example, kingfish is very popular in Australia. at restaurants and for recreational fishing. The eastern blue grouper is kind of an iconic emblem for the state of New South Wales. And then we also picked up a lot of these smaller cryptid species that maybe if you're doing a survey, you're maybe not looking for that because they're hiding in kelp glades or under rocks and so on. And at the bottom left here represents one of the endangered seahorse species that's endemic to Eastern Australia that we picked up in a lot of our samples in these locations. And using these data, we can start to have a bit of a play in looking at temporal trends. So through time, we can start to look at whether taxonomic richness peaks at certain times of year, whether there's seasonal effects. And if we look at two of the sites where we sampled over a 12-month period, we see that we tend to see peaks in summer, which tend to coincide with some of the spawning seasons for some of the animals and also some of the influx from tropical bits of Australia through strengthening of currents, and then some of the dips in the quiet times of winter. This applies also to coral reefs, so we've done a similar and equivalent study out at Lizard Island, so obviously a tropical reef on the Great Barrier Reef. And again, we're starting to see kind of dips in the diversity that we're picking up for things like bony fish and elasmobranchs, but also magnarians and sponges on the bottom panel in green here. So we're seeing, again, dips in winter and kind of peaks in summer times. Another big question that often comes up at conferences is, have we attained the Holy Grail? So can you get abundance of biomass data for all the animals that you're looking at? The answer is yes, but you have to be very careful about how it's applied. So I'm working with a group in freshwater estuarine environments in Australia that are trying to develop quantitative assays for three very important recreational and commercially important species in Australia, the murray, cod, golden perch, and mulloway. And to do this, they've taken a 5-tiered approach where they're very careful in starting with literature reviews and making primers that will only amplify these fish and no other fish in your sample. Then doing aquaria experiments where they're looking at manipulating different numbers of animals in aquaria, then stepping that up into mesocosms, so ponds where you have no biomass of fish, in particular outdoor ponds, and then finally applying them in the field. Some groups have started to mine biomass abundance inferences from metabarcoding assays, and it can be done. But again, it has to be done in a very careful approach because there's lots of things that can influence the number of reads, particularly when you're using these universal assays that amplify a lot of different animals. The last little bit of my talk here, I thought I'd talk a bit about some of the other approaches that we're using to record biodiversity. I have a very small research group, and when I was at the museum, it was a small research group. So we wanted to figure out ways that we can collect data in our environment without us having to go out every day, even though I'd love to go out in the field every day. Us having to go out and collect survey data in a lot of different places. And we've done this through citizen science. So we've set up small projects on the iNaturalist platform, which is, I'll talk about in a second, but essentially this particular project on the slide was getting people out into Southern Sydney Harbour to record marine biodiversity. And in this particular case, we had a number of dive agencies that were on board that were supporting our initiatives by providing vouchers and so on to some of the participants. So these data can be quite useful. This is one of the larger projects based on iNaturalist known as Australasian Fishes. So these are photos that are uploaded from anywhere in Australia and New Zealand. And we kind of downloaded the data set in the early days and looked at whether the records that we're getting for these particular fish, if you look at the map figured here, do they represent the actual known range for those particular species? And again, apologies that these are not tropical, these are temperate species, but you can see that we have a fairly good representation, the red dots being the citizen science records, and then the shading being the IUCN known ranges for those species. So there's a fairly close match there. For those of you not familiar with iNaturalist, it's a worldwide project now. It started about 10 years ago. It was started in America. It was a collaboration between National Geographic and the California Academy of Sciences, but it's really grown and exploded. You can see here, there's actually 193 million observations up there now. It's not just marine stuff. There's lots of terrestrial information on there. All you really need to participate is an underwater camera or a phone on land. And for the inclusion of a photo, it has to be a wild photo. You can't go to your aquarium at home and take a photo of the fish and put it up. And it also has to have a known date, a known location, and you have to assign some particular ID, but it's okay if you're not too confident in your ID for that particular animal or plant, because there's a whole community of people that will help you refine that identification. And so for those of you not familiar with it, yes, it is used in Taiwan. There's about close to 3 million observations at the moment. I did a quick search, and actually less than 1% are aquatic records. So most of them currently are plants, insects, birds, and things of that nature. So there's really some space for this to expand in the aquatic realm. Finally, these citizen science tools actually can be both adaptable and portable. It doesn't have to be a set project in a particular location. So I got the unique opportunity to go out on a research tourism vessel last November with Coral Expeditions, funded by Australian Geographic, where we had 43 able and willing tourists on board with us for 10 days that wanted to go out to Coral Reefs across the Great Barrier Reef in Australia and then out to the Coral Sea and learn a whole bunch of citizen science tools. And so we went out to some amazing places, saw really cool things on some of the centipedes, frisky turtles, for example. But yeah, it was a great platform to do this work. Here's a little map of all the different locations that we were able to get to over the 14-day cruise. And essentially we exposed them to a lot of different citizen science tools on board. The ones in green on the left are all the things that they actually got to try while they were on the vessel at all the different locations. And we ran a questionnaire at the end of the trip, and essentially they found that the two easiest for them and the two that gave them the most information the most quickly, so the most feedback, I guess, was using iNaturalist, so taking underwater photos and uploading them to that site. And then some of these eDNA tools that gave results back quite quickly with some of these commercial companies. And so here's some of the eDNA sampling out at sea with some of the guests that were on board. And very quickly we were able to generate these wheels of life, essentially, of all the different organisms that we picked up in our sampling. And you can see here that over the course of the 13 days that we were at sea, the citizens on board essentially uploaded 4,300 observations that represented 620 species. And we created a project under iNaturalist that was very easy for them to interact with. So I thought it was a great exercise in exploring all those different tools for tourists. I thought I'd kind of close off today just to explain some of the stuff that we got the pleasure of taking part in just before the conference. So Professor Colin Wen was hosting us out at New Island to do some sampling with Chris Goldie. We were looking at the cryptobenthic reef fish, which he's going to talk about tomorrow. And we were also able to do some eDNA sampling so we could compare those approaches with each other. And with that, I just want to thank everyone for coming, to the organizations that supported our participation today, and a lot of the organizers for the conference. And if there's time for questions, I'll happy to take some. So thank you very much.
Okay, I think we have a few minutes for a couple of questions for Joy. Any questions? Yes, please.
Thank you for amazing talk. I just wondered that, I'm going to talk about the potential challenge of using eDNA to quantify, for example, biomass, especially in a community approach. But I know that this is still kind of there's a long way to go in. But just my question is that, for example, different research groups, they may have different pipelines or something like that. So when we are saying that we want to use eDNA to quantify those community data, especially when pipelines are always different. So what are your suggestions? Whether we can, how we can, make some improvement to make this data more comparable across styles?
Yes, and that's one of the big issues right now. So the two areas where there's no real standardization is the lab process, where lots of people will use different volumes of water, filter sizes, and so on, or even assays in the lab that influence the results. And then the bioinformatic pipelines at the end, where you have to select different parameters that you're putting in, and maybe by changing one or two, you're actually changing the outcome quite a bit. I think what I've done is essentially whatever I'm doing, I've standardized going forward. So all of the studies that I do from now on, I basically follow that, so those are comparable. I think generally, most of it is comparable if you go back to the raw data that you use. So for example, someone else uses a different pipeline and publishes a paper, the data is supposed to be publicly accessible at that point, the raw data, so then you can then run your pipeline that you're interested in on that particular data. And there's lots of pipelines that are available now to explore the data in different ways. I would say for the abundance biomass question, you can do it on metabarcoding data, but it needs to be validated first. So you can't just pick up a new species and start that process and trust those data. A lot of people are using fisheries catch data to compare first, and then when they find close correlations between that, then going forward they use that approach. So it can be done, just needs to be done very carefully.
We have time for one more question. Anyone? Yes, please.
Hi, Joy. I'm quite interested about this citizen science part of your research. I think citizen science in Taiwan has become quite popular now, but most of the research or the result come from terrestrial animals. And in my group, we try to involve some fishermen, but most of them, for all different reasons, they refuse to report what kind of species they got or what's the location. So can you share your experience with us, like in Australia? Is either more researcher-based kind of citizen science, like for example, you approach it first, or the other way around, like local NGO or a group, they actually ask the scientists to involve or to lead them?
I think I would say that it's 80%, 10%, 10%. So the low participants would be recreational fisher people in Australia, which is a huge industry, but they're often hesitant to get involved. They just want to go out and get fish, and that's really where their interest lies. 10% is engaging scientists that maybe have data sets that are squirreled away somewhere that they didn't think there was a platform for them to submit to. But the big participants in these citizen science projects tend to be local dive clubs and snorkel groups, and starting to create those communities. So they're often the ones that are taking photos underwater, and they have stockpiles of photos going back 20 and 30 years sitting on the computer. And the nice thing about a platform like iNaturalist is it's very interactive. So that you join, you have a user ID, and there's lots of discussions between people in the community, but also with some of the experts in identification as well. So they feel like there is that tie between the science and the citizens. So I think, yes, targeting kind of local snorkel and dive groups is probably will be the biggest contributor.
Yeah, thank you so much, Joy. Let's give another big applause to Joy.
our first speakers, as I think all of you know him, Dr. Selin Tang from Academia in Biodiversity Research Center. And he, I think in this morning he already gave a very good talk, kind of trying to inspire us to work on the Coral Reef study. And now he is going to give us a story about Endocomonas. It's a coral bacteria, and Endocomonas and how function from the sulfur cycle, especially on the DMSP. So please just welcome Dr. Selin Tang.
Okay, good afternoon. Thank you, Sanhwa's kind introduction. And today I'm going to change a little bit my talk topic. Yeah, I didn't highlight the bacteria I wanted to talk about is Endocomonas. First of all, I want to introduce what is the DMSP, why we need to concern about the DMSP cycle in the coral horribile. Okay, so DMSP actually is a very small compound. The full name is dimethyl sulfone. And this molecule is one of the very crucial molecule in the global sulfur cycle. Okay, so here show you this short illustration. So basically, DMSP will produce mainly by the marine phytoplankton, and then metabolized by the bacteria, and then some of DMS will release into the air, and oxidize it, become a sulfide, and then can be facilitate the cloud formation. So the DMS has another name. It's called a climate cooling gas. Okay, and then it's an important molecule. This organic material compound. Actually, its physiological functions related to nutrients, almost like bioprotectant or antioxidant. After this degradation, the DMS can be used it's very strong this antioxidant, and then also, as I say, help the cloud formation, okay, to regulate the climate. And then, as I say, the biosynthesis of DMSP mainly produced by marine algae, and then the pathways is here. But one more information. Maybe I'm not sure if you are a bacteria person. Maybe you pay attention. But this is very, from my point of view, it's a very big breakthrough for the biosynthesis of DMSP. Actually, there's in the last 60 years, I think, around, the people found that actually bacteria play the key role of a synthesis of DMSP. Okay, from these two papers, actually they found that some bacteria actually enable to synthesize DMSP, and more importantly, as I say, phytoplankton is a major producer of DMSP, but they all need the light, and below the aquatic zone. All this is the huge, enormous water columns that bacteria produce DMSP. So I want to say DMSP mainly produced by bacteria in aquatic zone. You can imagine that this flux can be very huge. Yeah. And then this is another shock discovery. That's 2013. And then the coral animal, for the first time, people found that animal can enable synthesis of the DMSP. And then what they found, this is from the JCU group. And then this, the baby bone group. And then they found that this DMSP metabolism is very important for coral to combat this the thermal stress. In the thermal stress condition, the DMSP will degrade to DMS and will acrylate, which enable to clean up ROS. And the ROS, especially massively produced during the thermal stress. Okay, and so that's so important. And also, they look at the different temperature, 27 and 32. You can see this the DMSP production is increased at higher temperature. Okay. And so there's one very important pathway, metabolic chemical pathway, is DMSP is degraded by into the DMS and acrylated by many by the bacteria. So this is the first message I want to tell you. But then come back to the coral. In a coral, inside a coral bed, actually there are many bacteria which associate with DMSP degrading and DMS degrading. So then the question is, who played a role in the DMSP degradation metabolism in the coral bed? Of course, many of them participate. Who is the major player? So this is the question. Okay, when we want to find who is, then it's a big challenge actually. Why? Because this is human. Okay, if we use the microscopy eyes to look at it. And then actually in the gut microbiome, we can find 1,200 species. But in the coral, that's far more diverse, high diversity than what we thought. Yeah. For these cases, from the latest paper, 6,000 species in the coral. So many of them. And then they are also very dynamic. Over the time, and then you can see this dominant yellow color. Over the time, actually they're changing. That's the nature of coral microbiome. So then who played that role? So a long time ago, that's 2010, with Wu Chen, with Alan, they helped to find one Endospermos montifera. And then this is the first time they isolate this Endospermos from the stony coral. And then I found that these bacteria often appear in the coral we collect. And then later, with others, international scholars, we all work together and realize that actually Endospermos is very common, dominant bacteria in many different kinds of reef organisms. Yeah. And then also not only the species as well as the place, you can find all the many different places. Okay, so we try to complete this Endospermos montifera genome, and then we decipher the potential this symbiosis relationship between Endospermos and the coral. And then, so this bacteria has been annotated as a symbiotic, potential symbiotic. Okay. And then during the heat stress or the cold stress, you can see Endospermos community can shift very quickly to become the non-Endospermos dominant bacteria. Those bacteria is pathogenic this time. Okay. So over the many this last maybe more than 50 years, and a lot of the researchers try to figure out the role of Endospermos play in the corals. So including the DMSP metabolisms, nutrient supply, interaction with the symbiotic algae, secreting system, symbiosis feature, and also some disease or stress associated, also potential pathogen for antimicrobial compounds, all this. But one thing I want to highlight here is all this, most of the function actually is not, it doesn't have any the solid experiment to prove. Yeah. So it's mostly based on the elucidation or speculation. So we will move further. There's actually, I think early 2010 was the time, actually from David Bourne's lab, they speculate as they have a hypothesis. They hypothesize that the wasp should play an important role for this metabolism. But at that time, they haven't provided direct evidence. And then we are lucky, 2017, we isolated this new endozoognomus called endozoognomus acropora, obviously isolated from acropora species. And then we found the key gene, which is DDD gene. And then also look at the genome, we found the entire gene clusters existing there. The entire gene cluster of DMSP metabolism. And yeah, so we further confirmed that from the gene level and also the chemical level. Gene level, we look at the expression of the DDG gene. Along the time, we can see these genes. When we provide the DMSP, you can see although DMS, the gene expression is going up, and then at the same time, we look at the DMS concentration after 48 hours is increased. And also, because I'm thinking if this endozoognomus acropora is a very unique species in the canteen, then it doesn't mean too much to me, because that's maybe it's a very special cases. So we did some literature review, all the data collected from different research, and then realized actually this species actually quite dispersed in the different coral species, as well as different places, Red Sea, Western Australia, and then also the Great Barrier Reef and Taiwan. Okay, so yeah, and then for these particular small questions, we did a very tiny, small experiment. It's interesting to me because why we did this acropora actually has a better adaptation ability to survive in a different environment. So what I did is we went to the Penghu archipelago, and then inner bay and outer bay found the same species of coral, and then we split them into two parts. And then, so we do this reciprocal transplantation experiment. So we move the coral, some coral to inner bay, outer bay. Outer bay is a better condition in terms of environmental factors. Inner bay is a bit different, and the environment is harsher. So the result is interesting. When you outer bay move to inner bay, and very soon, within a month, this acropora become dominant in the microbiome. But then inner bay is another endozoognomus called endozoognomus pangenesis. When they move out to outer bay, very quickly it will be replaced. They cannot maintain it there. So endozoognomus acropora seems to have very strong adaptations ability from this indirect evidence. And also we conduct this owl-based activity test. And then for the lysate, pangenesis and acropora, it's actually pretty similar. But when you look at the supernatant, which is something secreted by the bacteria, you can see this acropora is very strong to these owl-based cleavage ability. Different from the pangenesis. Okay, and then we continue this isolation work. And this is another one we isolated recently. It's called Endozoic Ruscacia, AE. And Ruscacia also appear quite commonly in different sites. And it's quite big bacteria. And this genome is much bigger than Montifora. Yeah, Montifora is around 5 points something mega, and this is 7 point. But you can see this genome expansion is very interesting. They only compare other genomes here. You can see some this gene actually multiple replicated. Yeah, duplicated. Many copies. Sometimes several 20, 30, 40 copies. Yeah. Anyway, yeah, I'll give a name because it's a memory of Professor Yu's case. Okay. And at the same time, we also found they have this DMSP metabolism gene cluster. So you can see here, and this is acoporon. And this is monas is a very typical one. Okay. And then Pangolins, no. Mandibula, no. Okay. Yeah. So then, first of all, I try to analyze the activity of this DMS production. So this is Ruscacia, and this is 14. And this is Nigeria Atlantica, which has been reported. It's very this based, how is it, the most active species which is able to metabolize DMSP. However, if you look at this result, this is y-axis. Actually, compared to the Acropora or the Ruscacia, actually the activity is much lower than these two endozoic rocks. But of course, the Nigeria has own others activity because not only produce DMSP, but DMS, but also they have another demethylation pathway. So that way, that pathway won't produce DMS. Sorry. Okay. So we move further to synthesize the two type isotope. Where one is for sulfur, 34 isotope, stable isotope, and then for carbon, we put 30 carbons here. The main purpose of this experiment, we want to confirm this our observations. So we use the nanosim analysis, and then we can see this S34, because if this degradation happens, this DMS will release. And then the carbon is carbon 12, not carbon 13. So we can see that in 0 hour and 6 hour and 12 hour actually is going down. So all the DMS is released out. Yeah. But then for carbon, actually is keep. Yeah. First of all, I integrate and keep it. So this is confirmed again. This is our discovery. Okay. And furthermore, we look at the transcriptome, compare these two bacteria, and one interesting thing that we discover is this is a core pathway of the DMSP. Both bacteria, the gene, all activate, most of them. But if you look at the TCA cycle for the Acropora, actually, most of the gene actually is shut down, or I should say downregulated. But for the Ruscacia, it's upregulated, actually. So it turns out we speculate the DMSP for Acropora, this species, I think only the function is an antioxidant. But for Ruscacia, it can be a nutrient as well as an antioxidant. That's our speculation. Yeah. We need to confirm. But one more evidence I want to tell you. The endozoic metabolize the DMSP. This functional ecological role is not marginal. It's important in the coral horror. The result is that because we also check other genomes. This is uncultured technology, so they sequenced all the genomes from the Sesoko, from Japan, or other places. And then we all discovered this key gene, DDDD. Except that this one is from Red Sea. You can there's another one. It's a DDDP. And then many of them actually isolated from Acropora. So I want to say endozygomas actually is common, and the metabolic and this DMSP cleavage function is one of its major role in for life. So you can find more information in this paper. This is the paper we discovered the DMSP enabled to metabolize by endozygomas. And the funny is that year, they invited me to go to Magnetic Island in 2017. And then in that special meeting, comes up with the idea, maybe our bacteria enable to metabolize DMSP. So we check it very quickly, and we found it. And then we conduct all this, the work I just tell you. So this is also thank you. Good. Okay. And then, and the other one is we compare that, and so we can read more information from that paper. Okay. So take-home message is metabolism of DMS is a common function in endozygomas species. Endozygomas likely play a significant role for DMS breakdown and DMS production in the coral reef. And okay, I try to find one PhD student if I'm interested. Okay, so last slide, I want to say thank you for all these colleagues. Some of them become professor now. This is a professor, okay. And this is Wata's son. Okay, and this is another group. Okay, so thank you for your attention.
Okay, thank you very much for Dr. Tang's talk. It's very fruitful. To me, his talk is always inspiring me a lot. So, does anyone have a question?
Yeah, I agree. Tang's work is always inspiring. You learn something new every time. Great talk. Thank you very much. Maybe I missed this, but with Gatesii variant, is how widely distributed, how common, and how dominant is it? Can you tell me?
This particular species?
Yeah, that's good.
Yeah, some of the places like Western Australia, they have a sample is more than 75% of this the endozygomas are the Acropora protein.
Right. So it can be dominant. It's not just a minor component. Doesn't it have big consequences for the physiology of the whole organism?
I don't know yet. The consequence of what?
In terms of, if there's a protective function of the DMSP, then surely having an organism that completely breaks it down is going to have bigger physiological consequences.
Actually, I don't know, but recently I heard someone talk about some of the seabird also use the DMS as a crude, chemical crude, and also some organisms, marine organisms, also use this as a chemical crude to find something which they want. But I don't know these cases. Maybe we need to do, maybe find a marine the chemist, Tina, maybe can help. We can try to find out the flux, what's going on in there.
So, does anyone have a question?
I'm just curious. So the bacteria you isolated, I suppose you can culture in your lab.
And can they produce DMST in your cultivation conditions?
Oh, the bacteria I isolated cannot produce the DMSP, but can metabolize the DMSP and produce the DMS. DMS, yeah. And
So
then we
you
can
need to
measure
give them,
by GC, yes.
so you need to give them DMSP.
Yes, yes. So turns out this idea is Zimbabwean produce DMSP, coral produce DMSP. So those DMSP compound might be used by the ecosystem mollusks. And then in order to produce DMS, which might help to clean up the ROAs. All this is a hypothesis. Yeah. But can be like this kind of metabolic network.
Any question? Okay, I have a question. Yeah. So since now recently people are getting more analytical models become a family. Now it is not only a genus, it's become a family where we can find analytical models strands or species not in coral. So do you think do they among analytical models strands, do they compete or do they have any functional redundancy or do they can work with each other?
Yeah, that's a good question. Actually, I don't know, but someone asked is there any species diversity for endosperm mollusks? And I would say yes or no, because some species it looks like they are accommodated, always accommodate similar species in their body, but some can be changeable. Yeah. It depends on where. What I want to say is that, for example, you have Fossiliphora dalmatianis in the canteen, and the same species in the eastern Taiwan or the Green Island. But then ends up that you won't see endosperm not much, or maybe very few in the eastern, northeastern Taiwan. But you will see dominance in the canteen, or maybe Green Island. So some of them is environmental specific, and some of them is host specific. For all this information, actually, I think it's on top. The question is on top. Yeah, but that's very important. Yeah.
Okay, then following this question, sorry, I'm very interested in this topic. So as we know, we do a lot of bacteria, coral-associated bacteria, and finally in the end is try to use them as probiotics. Right? So far, how do you think about probiotics? Is this method, this kind of method of approach is also useful for coral restoration?
That's a good question. I saw one here, she was support. Very well. Anyway, I think it's yes or no, because as we know that the microbial community is changing all very dynamic, so quite dynamic, and then it depends on the place you pick up. Some place, I think if I use it as an example, maybe some place this kind of deployment to use them as a probiotics to improve their health, maybe not effective as what we want to see. But some place, for example, specific aquarium or the coral farm, they might be helpful. Yeah. So, but I'm looking forward to more this kind of stuff will appear. So yeah, let's see what's going on. But so far, the way to use the probiotic is actually not so. It's quite diverse. Some just take a coral out of water, please. Some just yeah. But think about it. If you want to apply the bacteria in the open sea, I don't think it's not so realistic.
So there's no question. Okay. So last thing, Dr. Tang again. Okay, on to the next talk. So next speaker is Professor Takaki from the Atmosphere and Ocean Research Institute, the University of Tokyo, and he will introduce about carotenoid producing bacteria in endosymbiotic immunogenic, which means this bacteria from symbiont. Yeah. So let's welcome him.
Thank you very much. Good afternoon, ladies and gentlemen. I'm Toshi Takagi from the University of Tokyo. And thank you for the opportunity to make my presentation today. Today, I will talk about micro-scale coral biome, especially focusing on the interaction between carotenoid-producing bacteria and symbiotic dinoflagellate. These are the bleached coral. As you know, mass coral bleaching is occurring in over 50 areas around the world, and the decline of the coral reef associated with coral bleaching is a serious problem. And when coral are exposed to environmental stress such as higher temperatures or high light, the endosymbiotic dinoflagellate produce reactive oxygen species, ROS, which cause the coral to release algae from the coral tissue or algae loss pigmentations, so resulting in the coral bleaching. Eventually, mass coral bleaching lead to the collapse of the coral reef ecosystem, like this photo. This is the figure of the ecosystem in symbiotic acid and the symbiotic dinoflagellate. And under normal condition and under normal temperatures, the PS2 produces electron from right and electron flow from PS2 to PS1, producing NADPH. And furthermore, NADPH is used for the carbon fixation in Calvin cycle. However, when the Calvin cycle is inhibited by higher temperatures, the electron react with oxygen to produce reactive oxygen species, ROS. And ROS inhibits the synthesis of proteins and the repair of PS2. Finally, the ROS accelerates photoinhibition. So the heat-dependent photoinhibition of PS2 leads to coral bleaching. And the coral and their associated microorganisms, including and the symbiotic dinoflagellate of the family of Symbiodiniaceae and fungi and bacteria virus comprise coral biomes. And the interaction between the coral host and Symbiodiniaceae or coral between coral and bacteria have been studied very well. However, the Symbiodiniaceae and bacteria interactions remain largely unexplored. The phytoplankton, including symbiotic dinoflagellate, segregate organic substrate into the region immediately surrounding individual cells and forming phytosphere. And it is like a coral mucus, and the exchange of metabolite and chemicals at this interface play important roles in the phytoplankton and bacteria interactions. The function of the phytosphere is like that of the rhizosphere of a plant. The Symbiodiniaceae cells also form the phytosphere. And our research has focused on the function of bacteria in the phytosphere of Symbiodiniaceae cells, and we have discovered that protect Symbiodiniaceae from environmental stress. Today, I would like to talk about mainly the two papers published in Enviro and Microbiology Spectrum. The first one is a paper published in Enviro, zeaxanthin producing bacteria isolated from outdoor phytosphere protect coral and symbiont from environmental stress. First, we isolated and cultured Dulcininium cells, formerly grade D, from coral, and then the bacterial community was manipulated with antibiotics. There were the result of 16S liposomal RNA gene amplicon sequencing data, and the control is no manipulation. ABX is antibiotic-treated algae and remove the bacteria family of Flavobacteriaceae. ABX plus GF1 is integrated with Flavobacteriaceae strain GF1 isolated from the control culture. Fluorescent in situ hybridization patient observation of the strain GF1 showed that it was present in the surrounding area of our cells, like this. And GF1 produces carotenoid zeaxanthin, which has high antioxidant and photoprotection activity. So we hypothesize this strain GF1 plays some functional role. So we carried out a stress exposure experiments. To examine the effect of the changed bacterial community on stress tolerance of the cultured Dulcininium, we exposed algae culture, aquaculture to salmon and light stress conditions. The ROS production measured using the ROS probe DCF-HDA, and the ABX group showed increased ROS and product under salmon stress condition and light stress condition, whereas no significant difference in the ROS production were observed and no stress condition. Next, we examined whether zeaxanthin supplementation to cultured Dulcininium cells can mitigate the oxidative damage induced by the salmon and light stress. Zeaxanthin supplementation in ABX culture resulted in significantly higher FVFM and lower ROS production. And under light stress condition, FVFM and ROS production had also a similar trend. These results indicated that zeaxanthin is effective in mitigating the salmon and light stress in Symbiodiniaceae. This is the first paper to report that Symbiodiniaceae also have a symbiotic bacteria with functional root like that of endocytomonas in coral. So second paper is published in Microbiology Spectrum, which lists the interaction between dinoflagellate and pigmented bacteria mitigate environmental stress. When we isolated Dulcininium cells from coral in embryos paper, we used F2 plus a Cas medium containing kanamycin, ampicillin, and streptomycin. This antibiotics mixture was proposed to Dr. Andrew Baker's laboratory from this paper for EG algal isolation from corals. So here we noticed that the bacterial quality of the Dulcininium isolated on F2 Cas medium was very simple, and they were composed mainly by 2-OTU alpha proteobacteria and Flavobacteriaceae. And these amplicon sequence data are from the paper Rosen et al. Environmental Microbiology Report 2018. And they isolated all Symbiodiniaceae cells without antibiotics treatment. And these data show the bacterial community composition of each Symbiodiniaceae grade A to grade D, and classified mainly at the bacteria genus level. So then we noticed that the bacterial community of Symbiodiniaceae should be more diverse. We thought that the simplicity of algal culture was due to the Cas antibiotics treatment. So next, I have a question. My question is, which promoted next research? Why carotenoid producing bacteria survive when coral and symbionts are treated with antibiotics? And is this a general phenomenon? So we observed whether carotenoid producing bacteria survive after antibiotics treatment. in other Symbiotic archaea cultures. And we use the chlorocopyr formalin, grade C, from our culture collection and Drusilinium cells isolated from coral in this study again with Cas antibiotics treatment. We carried out Cas antibiotics treatment of our cells in this procedure. Cas antibiotics treatment performed on agar medium for 50 days, and after that our colonies were picked and cultivated in F2 liquid medium without antibiotics. These are the colonies formed by the Symbiotic archaea, and the white edge are the phycosphere of the Symbiotic archaea. And the Symbiotic archaea cells form the colony on the agar plate and secreted an extracellular matrix forming phycosphere around the colonies like this photo. And an algal colony on the F2 Cas medium was picked and incubated in F2 liquid medium without antibiotics. And after that, algal cells were spread on the marine agar plate for bacterial cultivation. And these are the marine agar plate, the media with bacterial colonies. Surprisingly, in all algal cultures, carotenoid-producing bacteria increased after Cas antibiotics treatment. So we performed the bacterial community analysis of the microalgal culture before and after antibiotics treatment because some bacteria are unculturable on marine agar plates. And these results show the bacterial community analysis of the Symbiotic archaea cultures before and after antibiotics treatment. Left side, algal culture before antibiotics treatment. Left side, the ABX is after antibiotics treatment. The Drusilinium strain SGF isolated from the coral directly using Cas antibiotics treatment. The lower number show the percentage of carotenoid-producing bacteria in each algal cultures. As shown here, the carotenoid-producing bacteria in all algal cultures increased after Cas antibiotics treatment in amplicon sequence data also. So which also agree with the culture-dependent method in above. To further investigate interaction between Symbiotic archaea and bacteria, we isolated carotenoid-producing bacteria from antibiotic-treated Symbiotic archaea. Interestingly, we found bacteria genus Maribacter, which produce darker orange pigmentation from both ABX4077 from Caribbean Sea and ABX2466 from Okinawa. So we selected two Maribacter strains and Cytophagia roseatica strain DU1 producing pigment from the Drusilinium strain SGL also used. So next we perform antibiotic sensitivity test of 3 bacterial isolate and our strain GF1 from our previous paper in Enviro. All bacteria strains were able to grow on marine agar plate supplemented with kanamycin or marine agar plate supplemented with streptomycin. In contrast, all bacteria isolated were incapable of growing on marine agar plates supplemented with ampicillin. So result indicating that these strains possess low resistance against ampicillin. So next, we perform fish analysis to determine why bacteria survive after Cas antibiotics treatment. To reveal the localization of carotenoid-producing bacteria in 3 Symbiotic archaea cultures after antibiotics treatment, we perform fish analysis using the CY3 DNA probe targeting Probacteria archaea and Cytophagia. And red fluorescent was signal from bacteria. Red fluorescent signal was detected from bacteria on cell surface of Symbiotic archaea cells. And some algae were completely covered with the bacteria. For example, this cell and this cell, this cell, completely covered with the bacteria. So we conclude that symbiosis on the cell surface of algae is important for bacterial survival after Cas antibiotics treatment because phycosphere protect the bacteria from antibiotics. So next, we perform the functional analysis of Maribacter on the algal cells. Cradocopium sp strain NIES 4077 is before antibiotics treatment. ABX 4077 is after antibiotics treatment. After Cas antibiotics treatment, bacterium Maribacter is increased 8-fold based on the colony forming assay, and 100% of bacterial sequence was genus Maribacter. So next, we hypothesize this bacterial community manipulation could affect light stress tolerant Symbiotic assay. To examine this hypothesis, we expose microalgal culture to light stress for 14 days. And the red circle and yellow circles are squares light stress sample. And microbiome manipulated ABX 4077 named the host microalgal culture to maintain higher FVFM and the light stress condition compared to parent strain NIES 4077. And see that the Phlebobacter assay, genus Maribacter on algal cells, and Maribacter produce carotenoid including their sanction. And same as strain GF1 in biospectral. So carotenoid observed light approximately 380 to 500 nanometer from part of UV, blue, green, and yellow. So we conclude that the bacteria protect the symbiotic assay from the high light stress by producing carotenoid on the algal cell surface. The answer to my question, why carotenoid producing bacteria survive when coral and symptoms are treated with antibiotics? Symbiotic cells protect bacteria on the algal cell surface through phycosphere formation. And is this a general phenomenon? Yes, we see so carotenoid producing bacteria may predominant on the cell surface of the symbiotic assay. And we reported the first time a symbiotic relationship in which the symbiotic assay and the bacteria naturally reduced environmental stress. And the bacteria had a photoprotection activity against the algal cells and through carotenoid production. And on the other hand, the symbiotic assay protected bacteria from antibiotics through phycosphere formation. Then a new question lies, why carotenoid producing bacteria on the cell surface of symbiotic assay? Carotenoid producing bacteria protect the symbiotic assay and they act as sunscreen. So maybe symbiosis with carotenoid producing bacteria provide a survival advantage for algae. So we have discovered that carotenoid producing bacteria symbiosis with the symbiotic assay cells isolated from the corals. So I would like to propose a hypothesis that carotenoid producing bacteria are within the coral cells through the symbiosis with an adhesion to the algal phycosphere. Finally, I would like to talk about our latest research, complete manipulation of bacterial community in the symbiotic assay. The sphere of the symbiotic is the symbiotic assay itself, and attach the bacteria to the bacterium, the genus Maribacter. Let me skip the detailed procedure. We established the bacterial-free algae symbiotic assay strange and algal strange infected with a single bacterial strain, like this same image. So we inoculated bacteria-free symbiotic assay with 5 strains of carotenoid producing bacteria. The bubble plot showed the result of the bacterial community analysis of symbiotic assay strains. A larger sample indicated high abundance of bacteria. Before antibiotics treatment, more than 10 genera of bacteria were detected in both grade C and grade D. On the other hand, after manipulation of bacterial community in our cells, only infected bacteria were detected at the AS3 level in all samples. This result indicated a successful complete manipulation of the bacterial community of Symbiodini arcei strain. So next we perform heat stress exposure experiment of the algal strain. We selected two carotenoid-producing bacteria, GF1 and CD4077. Gray is the bacteria-free algae, and yellow is infected with Coral arcei, genus Lunicauda GF1, and red is the Coral arcei, manufacturer C4077. The manipulation of bacterial community result in the significantly higher FVFM and heat stress condition. The difference was especially large in grade C. For example, at day 7 under heat stress, the FVFM of bacterial-free strain decreased to around 0.1, whereas the algal strain with carotenoid-producing bacteria maintained FVFM to approximately 0.3. In grade B, algal strain with manibacter showed significantly higher FVFM when temperature was increased from 31 to 34 degrees Celsius. This result indicated that Coral arcei GF1 C4077 mitigate heat-dependent photoinhibition of Symbiodini arcei. And finally, we carried out carotenoid imaging by gamma microscopy. In this study, we used line scanning gamma microscopy produced by the co-author Dr. Kato in Tokushima University. And we used F2 medium for algal cultivation, but this medium does not contain a carbon source. So we observed whether the bacteria metabolize the organic matter produced by algal cells, and the bacteria actually produce zeaxanthin in algal cytosphere. After measuring the gamma spectra of each defined samples, we perform imaging of bacterial zeaxanthin. On the left side are bacteria-free Symbiodini arcei cells, and right side algae with bacteria, genus Malibacter. Red is the autofluorescence of Symbiodini arcei cells, and blue is the bacteria zeaxanthin. And no signal were detected from bacteria-free Symbiodini arcei. This is an enlarged image of algae with bacterium genus Malibacter. In optical image, this is the phytosphere around the algal cells. And in gamma image, we observe the bacteria around the algal cytosphere producing zeaxanthin, and the blue signal is zeaxanthin. As shown here, we have succeeded in visualizing the production of zeaxanthin in the phytosphere of Symbiodini arcei cells. In last, I would like to discuss how bacteria mitigate heat-dependent photoinhibition. Bacteria produce the carotenoid and observe light from a part of UVA, blue, green, and yellow. So we conclude that the bacteria on the algal phytosphere grow and block highlight like this and act as a sunscreen. And this function mitigates heat-dependent photoinhibition of Symbiodini arcei. In summary of my presentation, we isolated the carotenoid-producing bacteria from phytosphere of Symbiodini arcei through cast antibiotic treatment. And Symbiodini arcei protect bacteria from antibiotics through the phytosphere formation, while bacteria protected microalgal cells from environmental stress through carotenoid production. Carotenoids produced by the Coral arcei and the cytosphere of Symbiodini arcei mitigate heat-dependent photoinhibition. And I am also studying coral and bacterial interaction, focusing on the coral immune response against pathogenic bacteria. And a PhD student in my laboratory, Kako Aoyama, had a poster presentation on the P13. So please come to the poster, and I would like to discuss this research also. And this is all for my presentation. Thank you for your kind attention.
Thank you very much, Dr. Sensei. Any question?
Thanks for the talk. It's very great. It's fun. I want to ask about the fluorescence microscope photo.
Sorry.
Fluorescence microscope photo, 27th slide, number 27. This is the result from fluorescence irradiation.
Yes. Visual observation of bacteria on the harbor cells.
Yes, I'm curious about why some of the red dots are aggregate.
Yes.
It's like fragile. It's not always surround the symbiotic.
Yeah, it's a very good question. Some algae really cover, but some algae, pure bacteria, like this. So maybe depending on the cell cycle or something, the amount of bacteria will change.
Do you suspect the symbiotic almost died? You mean the symbiotic almost died because so the bacteria they don't always surround the symbiotic?
Yeah. Yeah, I think almost the algae have bacteria on the cell surface, but some bacteria, depending on the cell cycle, for example, the infragilate, remove the shells at most. So depending on the cell cycle, the bacteria amount will change.
Thanks. Thank you for your answer.
Any other question?
Yeah, I'm curious about the role of carotenoids. Well, Symbiodiniaceae, microalgae, they produce a lot of carotenoids by itself. Okay, so in natural environment, yeah, sure it's possible that some bacteria can produce carotenoids and cover on the surface of coral. However, it is really an issue of ratio. Yeah, if Symbiodiniaceae, I mean, inside the coral cells, if they can produce like 100, for example, 100 units of carotenoids, and on the coral surface, those bacteria can produce only 1 unit, then it's a 1 to 1 to, it's a 1% of protection. So in this case, that probably not very significant. My question is that have you ever measured how much carotenoids are on the coral surface and how much carotenoids on the coral surface and how much carotenoids produced by or content in these Symbiodiniaceae inside coral cells?
Thank you. So as you said, I should measure the amount of carotenoid in the coral and bacteria. But in this study, we established the non-measured carotenoid. So next step, we want to apply these techniques to the measuring the carotenoid in the coral and bacteria. Finally, I want to propose the ratio of the carotenoid in coral whole bio. Thank you.
Any questions? No. Okay, I have a question. So since you said that this kind of bacteria, bacteria, just live in the microsphere of the Symbiodiniaceae, so that means this bacteria also kind of symbiotic bacteria in corals.
You mean this bacteria detected as the animals?
Yeah, also live in the cell of the coral.
We strain GF1, the published in Bio, that one in the genus Mulicaula. This bacteria is common in the Symbiotidaceae. Every type of Symbiotidaceae and Mycobacter on the top position and microbial spectrum. This bacteria, the symbiosis with the pump algae. So I think this bacteria run in the shallow water area of the ocean.
Thank you.
Thank you.
So,
thank you for choosing talk. I have a small question. Probably like get during the talk. So there is a test for the heat stress. You test the heat stress after treating the bacteria to the algae. And looks like the quality, the Spirulina do not like the ability of protecting the Spirulina and photosynthetic efficiency for the quality is not so obvious. So why the quality is better, the quality is not so the heat protect ability of the bacteria looks much better in quality but not so well protected.
You mean the quality, the difference? Yeah. Because in my opinion, the grade half D have a high tolerance of heat stress. So the grade C is low tolerance of the heat stress. So we observed this change, and we couldn't observe the difference in grade B. So depend on our restrained heat tolerance. I think so.
Again, it's a great talk. I've got data over a lot of it, but I may be asking a really dumb question here, but right, these bacteria, you're proposing a symbiotic relationship between some of these bacteria and Symbiotidaceae. I can see the benefit to the Symbiotidaceae, but what's the benefit to the bacteria? What are they getting out of it?
Yeah, I think the Symbiotidaceae segregate organic substrate. So like the carbohydrate, so bacteria can metabolize and get food from the Symbiotidaceae cells.
Okay, so they're essentially competing with corals for carbon, is that right?
I think this benefit for only algae, and today talk mainly the two properties, Symbiotidaceae and bacteria. So I explained this slide. This is the beneficial future for the algae.
Yeah.
So,
but what about the bacteria? Where's the benefit?
Maybe this ability to change breaching sensitivity in the coral, I think. So, from next research, I want to draw this bacterial activity against the coral whole environment.
Thank you.
Thank you very much.
Okay, thank you. Thank you. Thanks, Dr. Our last speaker. So, our last speaker in this session is Sylvia Fontana. Introduce us the seaweed diversification driven by Taiwan's emergency and social current. So, let's welcome her.
I'm talking about dispersal of species of red algae. So this is my target species. It was initially described as Lolaxara elegans. I say initially, but we'll see later why. So So it was described in 1935 from Kenting in south of Taiwan. This is the original description, which is only drawn like this, so it was a little bit difficult to match the original description with our new sampling, but it was described from Kenting together with in Taiwan together with another species which is called Galaxara latifolia. They are very similar to each other. So this species are classified marine red algae, and one of their characteristics is their tail is flat, and this is again very important data for identification. Another important thing, so generally red algae, if they are limited dispersal activity. So because they don't have any floating stage, and also they don't have flagella, so their dispersal is they disperse less distance compared to other marine organisms. So it's interesting to study their dispersal because it's generally more limited. The distribution of this species now is in the Northwest Pacific Ocean, so around Taiwan, Japan, and the Philippines. And also it's kind of shallow, so we can find in the 3-meter deep rocky shore. And this is also important because it helps us identify compared to other species, and also it's easier to. So this is the picture. Another characteristic is that the gametophyte and sporophyte stage, we have two life stages. They look the same, but at the cross section, we see that the cortex is actually different. The type of epidermal cell can be easily distinguished. So this species is morphologically similar to other two that you can find in Taiwan, which are called Trichomaria marginata and Galaxara latifolia. These are their photo, because when we collect in Taiwan, we find this type of algae which are flattened. Initially, if you are not expert, you don't know which one of it, but there are some way to identify them. So, first of all, our target species, elegans, is kind of brighter in color, but that's not always useful when you're underwater. And it's also found a little bit shallow area, but again, it's not 100% that we can often find. Like, it's found below 3 meters, the other are found around 5 meters. Their appearance is very similar. So to distinguish those species and be sure which one is our target species, we also did some morphological analysis. In this case, we take the branch width, and you can see that elegans, which is their width, which is this one, is much smaller. So we can also identify them. We also did some cell measurement to see if there is some morphological difference which is significant between these three species. And so this is the gametophyte stage. There are many cells, of course, but then we find that only the spine cell and the first layer of the epidermal cell are consistent enough in the same species. Other cells, there's too much interspecies variability. But again, in the gametophyte stage, we don't see so much difference, only that marginata has a wide epidermal cell. There is more difference in the tetrasporophyte stage. We divide our measurement between the middle and the edge of the branch, to be more precise. So this is how we look at the surface of the tetrasporophyte. We have the last layer of the epidermal cell and this kind of stalk, and another cell which is attached to it, we call outer cortical cell. Again, we can see some difference. So if you have a lot of information, you can, with the measurement,
gene, and is a phylogeny of this Dictomaria marginata complex. Why it's called marginata complex is because historically these species, because they are all flat and they all look similar, when they were collected they were all called marginata. And like more recently, not very recently, but maybe like last 15 years, there are more and more species described. So now it became complex, with some of them still called marginata. And we also perform species delimitation based on the two markers, with different methods, which is called GMOC RPDT. I'm not going into the detail of this method, but basically one box is supposed to be one species. But okay. So one thing, so you can see that the species which is called Dictomaria marginata is actually polyphyletic. So these are the species that were called marginata originally, but they were described recently. So probably this also need a better description, but we don't know the original one. The original one is in the Caribbean. This also has a representative from the Caribbean. So possibly there is one species called marginata, and we don't know which one it is. Then another things to consider, and those species are all restricted geographically. So like this coral intermediate, like found in Galapagos, one is only found in South Africa, and our target species is called Galaxora elegans, is found around Taiwan, Japan, and Philippines, and like the other species which we are focusing in Taiwan is called Latifolia, is only found in Taiwan. What you can say about this species complex, which is still need a better description of the species, and also we don't know the relationship between them. So the clades are very well defined, but we don't know, maybe because the marker is not powerful enough, or maybe there is another reason, such as like species radiation. So if we check our target species, and the other one which is found in Taiwan, well, they are called Galaxora initially, but clearly they are part of this Dictomaria complex. So we need to transfer that, to change the name into Dictomaria elegans and Dictomaria latifolia. So this is clearly because this genus Dictomaria was actually proposed, or like re-proposed actually, after that these two species were described. Were described in 1935, and at that time they were all called Galaxora, and then the Dictomaria genus was established later. So after 1935, maybe nobody worked on them, so they're still not being transferred. Well, these are the species that we recently described. So, okay. So now we established like the baseline of the species. From now on, we call it Dictomaria elegans because it's really part of the genus. And we can see there are actually three clades, which actually 1, 2, 3. The relationship between the clades are more defined here. So that's clade 3, which is kind of basal to 1 and 2. But we don't dare to call it 3 different species because, okay, if you see the fox 1 species identification, look like there are 3 different species. 1, 2, 3. But then the plastic marker is not concordant. So clearly there are 3 lineages. We didn't establish a species status for those 3 lineages because the markers are not concordant with each other. And maybe we need a different, maybe an intramarker, or we need a more, maybe genome, we need more information. Okay, but for now, yes, we did add one more marker, which is again mitochondrial COX-2-3. That is more valuable because it's an intergenic region. And we take some representative of each lineage. And we can see that actually the relationships between the lineages are not so clear. They are pretty clear. The lineage 1 is found in Taiwan and Philippines, lineage 2, Taiwan and Japan, and lineage 3 only in the Philippines. But the routing is still not 100% sure. Why I show you this? So this is, because our sample size initially was very small, so we increased the sample size, which is like more than 100 species, more than 100 samples, and we decided to use this COP2 to increase the sample size because it's the most variable. The results are similar to the result we get with a lower sample size utilizing COP1. Basically, the lineage 1 is the less diverse. It has one main haplotype and a few other closer haplotypes. And the lineage 2 has more diversity. And surprisingly, lineage 3 is actually kind of part of lineage 2 according to mitochondrial data. According to plastic data, is basal to the 3 other lineages. So this is also probably some case of incomplete lineage sorting, or we need to use more data to know what's the relationship between lineage 2 and 3, especially between lineage 3 and the other 2. To show you the difference, this is the same, the haplotype network based on the plastic marker. And they are completely divided into 3 different lineages. And I wanted to show you that the lineage 3 is the southernmost from Sorsogon, which is like east of the Philippines. And then the northernmost specimen we have is from Japan, around Tokyo. And I will not show you later because unfortunately this is not our specimen. It's a sequence that you find in JPEG. So we could not obtain the other part of the specimen. But we have a specimen which is in Japan. And it's the northernmost and it's like belong to image 4. This is the same haplotype, but we put it on a map so you can see the geographic distribution better. So basically, lineage 1 is found only in the south of Taiwan until Taiwan, and in the north of the Philippines. So it's kind of restricted here. I mean, this makes sense. We don't know why we find also a specimen in the north of the Philippines. And then we have the lineage 2, which is in the Batanes and Lani, Litao, north of Taiwan, and also we have sample in Japan, which is supposed to be here, but I cannot show you because we don't have copies. And also we have one specimen in Dongsha. The type locality is here, so we don't know exactly the coordinate because they were not reported in the original description. And this is Sorsogon, where the lineage 3 is, so it's very south. We do have samples from this area, and we couldn't find this species, but maybe the sampling need to. So this makes more sense if we check. First of all, we check the bigger map with also the sample from Japan, which I was talking about, and we can show you exactly what it was. No, I say talking about just to say nothing. And we also need to consider, of course, the current system. We have the North Equatorial current, which is bifurcated into the Kuroshio current and southern version is Mindanao current. So the lineage 3 is kind of when this happened. So we don't know if the lineage 3 is being picked up by the Kuroshio current or not, because this specific latitude, according to some publication or sometimes, because the North Equatorial current is not exactly all the time hitting the same latitude. So it can be picked up in the Kuroshio current, but also like sometimes the Kuroshio current is a little bit northern. So it's an interesting placement, and we can discuss about it. So the lineage 2, which is the green one, you can see that it really follows the path of the Kuroshio current, including this intrusion into the South China Sea. Notice that in the intrusion does not touch the north of the Philippines and does not touch directly the south of Taiwan, but it can reach long shot. So this is also interesting, and it shows that the distribution of lineage 2 is an classical distribution of a species which is being transported all the way up north by the coastal current. But lineage 1 is not, it just stays in the south of Taiwan. Again, we have other species which are not algae that they can still be in the south of Taiwan and be transported, but keep in mind that these species have a very lower dispersality. Of course, we also want to see there is some environmental factor between the lineage 1 and 2. We didn't use lineage 3 for this analysis because there is only one location and we have fewer samples. So we want to see if the lineage 1 and 2 have some adaptation to some environmental factors. So this TISCR analysis includes 18, 14 environmental factor, and we can see this. You already see in the map where the location there is a lot of overlap, but there is this significant difference between the environmental factor in the two locations, which is clearly driven by the outlier, like this is the northernmost and this is the southernmost, but still there is some difference, so we can see that the lineage 2, which is the one which is transported by the coastal current, the location where we find have a higher current speed, oxygen concentration, and nutrient and salinity. And while the lineage 2, the lineage 1, which is the one we find in south of Taiwan, those locations are generally higher temperature. So how do we interpret this data? So it's kind of a little bit obvious that lineage 1 is finding higher temperature because it's generally found in the south, and we have lineage 2 a specimen from Japan, which the temperature will be much lower. So this is also driven by this outlier. And then the lineage 2, which is the one really on the part of the coastal current, in those locations we have higher current speed, obviously, oxygen, and also higher nutrient. This is probably because during on the part of the coastal current there is some aquarium, and there can be some nutrient picked up. However, this is just the fact that there is some environmental difference between the locations, which are mainly lineage 1 and 2, but we don't know if actually there is adaptation or the two lineages are mainly driven by the current and the historical factor. So another data that we gathered to analyze this is we do this analysis, which is called isolation with migration, to find the divergence sign between lineage 1 and 2. Again, we didn't use lineage 3 because there is only one haplotype and the diversity was not enough to get solid data. But so this analysis showed that the lineage 1 and 2 diverged around 3.7 million years ago, which is kind of old for just population. So we think there can also be some sort of incipient speciation based on the fact that they're very old and there is no sympathetic overlapping between the two. And this is the Bayesian skyline plot, and we show the population dynamics. So basically, both of the population are slightly increasing, but there is no gene flow between them. So there is the separate, like slightly increase of the population. Okay. Conclusion. So conclusion in two parts, because first I talked about the diatomeria phylogeny, and so we use this genome diatomeria and we analyze the diatomeria complex. But this already gives us some trouble because first of all the species relationship within this complex are really unresolved. We can see that there are really different species, but we don't know the relationship between them. So it's actually difficult to pick up like an outcrop for our species, and probably we need to increase
increase the sequence gene or the sample size to know better about this. So one thing that we can say for sure, there are many endemisms. So most of the species found by our analysis, but also described by previous research, are very restricted to only one location. So I already talked about that the red algae have restricted dispersal, so this is possibly the reason. Of course, there is also some species which is not restricted. So how do we discuss that? There is a circumtropical species. However, because we didn't work in detail into this circumtropical species, it's possible that it's actually composed by multiple prickly species, and we haven't analyzed yet. And because actually there is the circumtropical by mainly restricted to the Caribbean and to the Pacific Ocean, so it's also possible that there is some human-made influence, and we have this artifact of dispersal. And of course, I haven't put it here, but we have the situation that our species target was initially named Calypsora, but we find out it's actually Dicomeria. So naming the species is already we need to name the species before we can analyze that dispersal. So in general, there is a need for a better revision, species characterization within this genus, within this complex, but this can be in general for other species or other species complex which have this situation. So conclusion 2 is more about our target species. So we find 3 lineages. The 3 lineages, however, are completely allopatric, so you can never find them in the same location, despite a really extensive sampling, especially in some areas. So it's possible there is an equivocally sequence speciation. So the lineage B is the more perplexing, the one from South Togo, because it's really the start of the crucial current. So we don't, it can be an ancestral species or a sister lineage for species. We see that the lineage 1 and 2 are separated 3.7 million years ago, which is like after the formation of Taiwan. So our conclusion is that they are mainly separated because of the island formation and the forest and just the first colonization, and there's because of the current and the dispersal ability is not great, so they do see a complete separation of the lineages. So the lineage 2 has high diversity and is clearly dispersed through the crucial current, but it's not the case for lineage 1, which is interesting. So it's kind of isolated in the south. Again, if you see the current system, there is a presence of eddies around the area, and the intrusion and this eddies doesn't reach the down shadow of the South China Sea, so that's why we see a more isolated situation for the lineage 1. So this is our reconstruction of the population dynamics with the data we have so far, and of the species. Thank you.
So neither, because of time, so neither we can offer maybe 1 or 2 questions. Thank you. I'm just curious, like, do they often you can observe the sexual reproduction of this species? Because it seems like even though in other, for example, in other species, it might be easy to see a picture of maybe a hybridization or something, but in your case, it seems like they have been isolated for quite a long time. So, do you think, like, because I don't know this species, so how do you think in terms of their sexual reproduction
Like, is it really limited in the local area only, or it is hard for the sexual reproduction to disperse to a long distance? Because you see in the brochure, you also have a similar species from Taiwan, so it's kind of like a long distance to the end of the brochure. So I'm just curious, like, how can you explain this?
So in my opinion, first of all, we have there is gametophyte and sporophyte, and the gametophyte is a little more rare. Well, we can only find with the cross section, but because we have a big sample size and we do a lot of cross section, it's a little more difficult to find the gametophyte. But yeah, the sexual reproduction happens. Also for the collection, which I'm less expert because I rarely work for collection, but if you look at the population, each population is isolated in a specific area. So I think it happens like if you go to a specific location, you will find a lot of colonies which are maybe there is some sexual reproduction within, but less. And I think this big distance is just done by longer time and once in a while it happens. But the majority of the time is just one location, some sexual reproduction, but mainly a sexual reproduction of many colonies. We try when we collect, we try to if you go to one location to swim a lot, because probably if you pick up the colony close to each other, they will be grown. So this is I think, yeah, it happened, but it's less frequent. But again, this is my opinion from the sampling of the women.
Okay, so because of time, so thanks Fiona again. The next presentation. So outside, there's some food, and so the break time will be 30 minutes. So please come back to this room or room 103 or 135. Thank you.
So hello everyone, welcome back to the sections. And the first one will be introduced by Professor Shinato to talk you the uncover the mystery and coral reef. So let's welcome Professor Shinato.
Okay, so thank you very much. First of all, I would like to thank the organizers to invite me to this conference. And I'm very happy. I'm very happy because I have many friends and many friends in Taiwan. So actually, this is my third time visit Taiwan in last half year. Actually, already I visited Taiwan many times, and I met many friends. And also, not here, but my PhD supervisor here, but he goes somewhere. Anyway, it's okay. Okay, so today, basically I graduated David Miller Lab a long time ago. My background is more zoology, coral biology, and recently genomics. So today I'd like to talk about my genomic study, focusing on the symbiosis, population genomics, and eDNA. So, because this is a Coral Reef conference, so I think most of you understand what are coral reefs, how coral reefs are important. The coral reefs have a rich biodiversity, and coral have symbiont, and so on. So, this time I will not talk this kind of, I'm going to skip everything. So I would like to start from this slide. Okay, so as you see, genome contains all biological information. So, of course, coral is animals, of course, multicellular animals. So they have nucleus, and in the nucleus there's a chromosome and DNA, and all the genes are stored in the DNA. So, of course, genome contains all those biological information, so it means coral genome means all the biological information that makes up a coral. So if we have coral genome data, we could get a lot of biological information of corals. So this is our motivation to start coral genomics study. And then after the first sequence of the coral genome in 2011, as David explained, we keep sequencing coral genome. So these are the example of the species that we sequence the whole genome. So mainly genus Acropora and some Montipora, Asteropora, and Proites, and probably another one coming soon, hopefully. And so we are keeping sequence the genome of coral, and also we sequence symbiogenous culture strains. So this So after today, you guys listen to Director John's wonderful scene. Probably you will think that, oh, there's another one I want to sing, but sorry that I won't do that. Yeah, my voice is terrible. Yeah. So first of all, I would like to thank the organizers for giving me this opportunity to share our work on coral ecosystem soundscape. So my background was marine bioacoustics, especially focused on marine mammals. But right now in Sumika, I work on the soundscape. Yeah, and focus on marine ecosystem dynamics. That means I use underwater sounds to observe all kinds of ecological processes. Okay, so I guess here everyone knows that global coral ecosystems are threatened by climatic and exothermic stressors, which have caused widespread habitat degradation. But when we talk about habitat degradation, we usually think about indicators such as coral coverage or reef complexity. But one of the habitat dimensions often missed, especially by marine ecologists, is acoustics, like underwater sound. So for example, this is the underwater sound taken at the same place at Wave 1 Lüdao, the Mesophotic site. Okay, so that's not some ducks or chicks, that's a fish are making noise. And also you hear lots of bubble-like noise that's from probably crustaceans or salmon ships. So why the coral reef or coral ecosystem are so noisy? Because there are many animals are producing sound, not coral to make sound, but many other organisms like crustaceans, yeah, or denser fish. Okay, so they will make this kind of noise during their territorial, administrative, or reproductive behaviors. So in general, you can hear crustacean snaps in high frequency range, and most of the land sounds just like bubble noise. And in low frequency range, you will hear lots of transient calls this kind of signals. Yeah. And these biological signals are very typical for coral ecosystems. And right now there are many studies showing that these signals are kind of important habitat cue for larval recruitment. For example, these studies they use a choice chamber experiment to demonstrate that coral larvae they can perceive the acoustic heterogeneity and then use this heterogeneity to locate their habitats. Yeah, and also similar studies have been performed on fish larvae or comical other taxa. Yeah. And also recent field studies also show that between healthy reef and degraded reef, soundscape also show very different conditions, especially in terms of the phonic richness or acoustic diversity. So definitely sound can be used as an important habitat indicator for coral ecosystem. Okay. So just in case that you are not very sure about what is the term called soundscape, soundscape is a mixture of sounds from environmental, biological, and anthropogenic sources. So for example, the case study we conducted in Okinawa, that we have we run some kind of long-term monitoring at the 20-meter depth deep for about 2.5 years, and then we're trying to use machine learning algorithm to separate biological corals, shipping noise, and wave noise, and then so that we can visualize the intensity change across 24 hours and between different seasons and years. So for example, during the summer nighttime, we can hear this kind of biological chorus. That's lots of reef fish are making calls during spawning season. And also during national holiday like the Golden Week in Japan, we know that's a very busy time for tourists, and we can hear lots of shipping noise. Yeah, and also during typhoon season, you can hear intensive waves. So to me, these are all because we can just deploy one recorder there and then just pick up all this information. All we need to do is just try to solve this kind of complicated interactions. Yeah. Okay. So since 2018, when I still was working in Gen State, we initiated this ocean biodiversity listening project, trying to cover all kinds of marine ecosystems, from shallow water to deep sea. And in Taiwan right now, one of our focus is coral reef ecosystem, including northeastern Taiwan, Lüdao, and Xiaoliucheng in southern Taiwan.
So what we're trying to do is to deploy like a long-term C4 recorders to capture the temporal variation, and also we conduct both like surveys or do like a snorkeling or like a kayaking to mapping the spatial variation of underwater soundscape. And our goal is to try to establish a baseline of so-called soundscape diversity in coral ecosystems. And then we want to, like through time, we want to know how environmental change shape underwater soundscape, and also how human activities degrade the soundscape, so that we can better predict the soundscape change in different kinds of scenarios, and then suggest conservation efforts. Okay, so first of all, I would like to talk about soundscape diversity. So right now, we in these 3 regions, we have established about 10 recording sites. Of course, we still have 1 or 2 recorders in South China Sea, but that will not cover in this presentation. Yeah. So the reason we choose these 3 locations because they have very different weather pattern. Northern Taiwan have very subject to strong seasonal variation in seawater temperature, and also in the winter time, a strong northeastern monsoon. Yeah. And although Xiaoyouqiu and the Lüdao, they are both in tropical region, but the difference between these 2 islands is mainly during the winter time. Yeah, Lüdao is still subject to very strong northeastern monsoon, so lots of intensive waves. Yeah, and also Xiaoyouqiu, because sheltered from the central mountain of Taiwan, so even in the winter time, the water there is still relatively calm, and then the environment there is very stable. Yeah, so we have all these 10 recording sites covering different water depths and different areas, and 4 or 5 sites were established in the middle of 2020. That's the time when I back to Sinica, and then right now we have at least 9 sites are still running, and then we hope that we can do this as possible as we can, as long as the budget is not shut down or something like that. Okay. So the way we deal with this massive data set is that we don't actually listen to all of them. We look at their variation. The way we look at their variation is through some kind of signal processing technique or statistic to look at the sound intensity in different frequencies, for example, along this y-axis and along the time axis. Yeah. So for example, here we can use a median possible density from each short audio clip to visualize some mass phenomena. For example, the Australian snap or nighttime fish course, they are all very intense during the nighttime. And even in this case, in the weekend, we can detect lots of human-made noise, mostly come from diving activity. And also during typhoon warning period, we can detect very intense wave noise in low frequency range. And for transient signal like fish calls, we can also use other statistics to visualize their occurrence. Yeah. So depends on what kind of sound source you want to visualize. There may be a way you can figure it out and then choose a specific frequency range to measure their intensity or diversity in frequency domain. Okay. So let us look at the soundscape diversity in Taiwan. Yeah, so each panel shows different recording size, and here this small region shows that the average daily spectrogram. So here the X shows the 24 hours, and the Y shows the frequency. And each small part here represents an average of 2 months period. Yeah, so here is basically just the soundscape phonology specific to different sites. So to me, the most interesting part is that for the sites in northeastern Taiwan, you can see that this one in Chaojing, during the summertime, the high frequency range is very intense because of the high level of crustacean snaps. Yeah, and this one in deep water in the summertime, that during night after sunset and before the sunrise, we can detect lots of low frequency fish calls. Yeah. But if you move to the Lüdao and the Xiaoyouqiu, this kind of seasonal phenomenon is not there. You cannot see this kind of seasonal pattern. For example, in Sanfu, it's very, very stable. Yeah, and also one of the very interesting patterns that in subtropical region we detect lots of fish call in the nighttime, but in Guiwan or in Xiaoyouqiu, we detect more fish calls in the daytime. Yeah, so there's some kind of interesting pattern behind this soundscape diversity. So if we use the UMAP technique very similar to principal component analysis or the MDS, this kind of dimensional reduction actually tells us that the soundscape diversity right now we observe is mainly driven by sites rather than by seasons. So that means if you have a chance to dive in different, like a coral ecosystem, you can hear different music. Yeah. So this is kind of diversity is actually quite interesting, and that's some kind of natural resource we should also protect. Okay. So as a soundscape project, I'm really interested what kind of mechanisms are like driving this soundscape diversity. So for example, here we can try to calculate the average of a monthly average on this kind of diversity map. So we can see that, like again, different sites occupy different areas, and in northeastern Taiwan, they have much wider variation, represents the seasonal variation. Yeah, and on this like diversity map, we can see that each green there's like typical like a daily spectrum, what it looks like. Yeah, and we found that there's a strong like a pattern along this direction. It's like driven by the snap intensity. And if you look at the right side, this like a yellow size are mostly like a deeper size, greater than like a 15 meter or like a 20 meter. For example, this one in Chaojing and the 4 sites in Lüdao. Yeah. And on the diversity map, you can see most of the like monthly average are located at the left part on this map. That means that deeper water sites, they have like lower constellation snap intensity. And this kind of pattern is quite consistent with our previous publication in Okinawa. So like snaps are very intense in shallow waters, but if you go deeper, then like you can hear a weaker like snap intensity. Yeah. And another direction is along this one. Like we found that like for example in Lüdao or like Xiaoliuqiu, that like most of fish calls were detected in the daytime, but in northeastern Taiwan, most of fish calls were detected in the nighttime. And we think that this may be related to the latitudinal effect on the fish assemblages, and possibly because of their like habitat. Yeah. But right now we are still not very sure about this because, for example, in Lobster Cave, this is also from Xiaoliuqiu, but we rarely detect fish calls. But like if you have visited there, you know that that place is highly degraded by human activities. Yeah, so probably this is not an exception. This is just simply showing that the habitat is not very good there. Okay. So right now, in order to understand like this kind of habitat divergence, so our team also trying to integrate soundscape and environmental DNA metabarcoding to understand how fish assemblage affect our like the sound diversity patterns. So like my student Wei Zhen, she just finished her presentation in an hour, and I hope she's doing a good job. Yeah. And anyway, like her result basically shows that fish sound diversity very different between photic and mesophotic reefs. And using eDNA, we can kind of show that Pomacentridae or Pomacentridae may be the families responsible for this kind of divergence. So right now we are trying to expand this kind of technical integration to our recording site so that we can better understand the habitat divergence. Okay. So right now I will talk about the environmental fluctuation. Okay. So if we look at the 3 long-term sites in northern Taiwan, Lüdao, and Xiaoliuqiu, the upper part shows the seasonal change in seawater temperature, and the lower part shows the seasonal change of like cross-section snap intensity. And you can see that only the northeastern Taiwan that have a seasonal changing pattern closely related to the water temperature. And we know that like, because this area is a subtropical region, that so the biological activity basically driven by the seawater temperature. Yeah. But in Guiwan or like the Lobster Cave in Lüdao, Xiaoliuqiu, although the seawater temperature still has some kind of seasonality, but it seems like they don't have a really apparent impact on the soundscape. Yeah. But instead, we can see some kind of variation between years, which is unexplained by the seawater temperature. So let us look at the 2 sites like in Lüdao, the south part of Lüdao. Yeah. So we have like recording site from 20 meter and 40 meter side, and they both show a very interesting patterns that weaker sound intensity in the fall of 2020, and then gradually recover in 2021, and then remain stable in the rest of the years. So if you go check the temperature, like history, in recent years, you will know that in 2020 we have a mass coral bleaching in southern Taiwan. And so this is the photo taken by Zhao Yang and nearby our recording site. So even I think in Mesozoic part, there's still some part of the population may suffer from bleaching impact. Yeah. So this kind of soundscape recovery may suggest the impact of heat stress on coral ecosystems. And certainly that we can use this kind of soundscape stuff to kind of track what's the dynamics of coral ecosystem after the bleaching impact. Okay, so this is another case in Xiaoyouqiu that we have recording site at the eastern part of this island and the western part of this island. And also we do the seasonal on-board surveys across this island so that we can not only track their temporal variation but also map their spatial variation. Yeah. So because of the some budget issues, so we didn't really initiate these two recording sites in the summer of 2020. So we cannot really see that kind of pattern as in what we have observed in Yunnan. But in 2022, again, we see another heat stress. But this time, the corals are still kind of okay, not in a mass bleaching condition. So we do not really see that kind of a major fluctuation in terms of sunscape. But it is quite interesting that in 2023, the heat stress here, and then there's a series of typhoons. They are all very close to Xiaoyouqiu. And then we found that both recording sites show the substantial degradation in sun intensity. So if we compare the sun distribution map before, in the middle of 2023 and close to the fall of 2023, especially after a series of typhoons, you can see that the difference, the brown color suggests the areas that have been suffered from extensive degradation in coral ecosystem soundscape. Yeah. So I think this is as a methodological development, I think this is quite interesting because this shows that the potential we can listen to the sounds and then visualize where are the reef being impacted. But also when I look at this result, I feel quite unfortunate that this kind of case actually happens. Yeah, and showing the degradation of the ecosystem. Okay. So, to further into the typhoon impact, if we look at the high resolution time series, for example, the 24 hours continuous recording, then we will see that this long-term recording site in northeastern Taiwan during the typhoon Mayfar in 2022. During typhoon period, the low frequency nodes actually suggest that the wave is very intense when typhoon is very close to this area. And then our in situ measurement actually shows that the degradation of soundscapes during the wavy period. But if we compare to the model prediction, if we assume there's no typhoon disturbance, for example, we use this part of data to build a model so that we can predict the time series, then we can actually see this kind of loss of acoustic activity that may suggest the soundscape degradation and the recovery process. Okay, so if you are interested in this kind of data set, please stop by poster number 1 and Sharon will be happy to talk with you. Yeah, well, I guess. Okay. Okay, and then right now we're trying to sample all this typhoon impact data. Yeah, so for example, from 2021 to 2023, we have accumulated a number of typhoon events. And in the beginning, I kind of hesitate to leave the recorder there because, the recorders are very easy to just disappear after typhoons. Yeah, but luckily they are all survived. So thanks God. Yeah. So right now we can see this kind of degradation and the recovery process across the 3 long-term recording sites. They are all very similar. But the interesting part is the scale of disturbance varies by typhoon events and also location. I don't know why, but in northeastern Taiwan, the scale of disturbance seems quite high, no matter whether the typhoon is very close to the recording site or far away from the recording site. As long as there's a heavy rainfall, then the soundscape will be degraded. Yeah.
But in tropical regions like Xiaolioqiu or Lüdao, the scale of disturbance is usually like kind of not very high. Yeah, but in one case, for example, this 2023 Doksuri, this like a wrong trajectory here. This one is very unique because it directly, go from the southern Taiwan and then into a Taiwan Strait. So most of the typhoon impact were not sheltered by central mountain of Taiwan, and then that caused a very extensive impact on the reef soundscape. Yeah, so this is the interesting phenomenon that right now we observe through this long-term monitoring. And the most interesting part is for Lüdao. Even though there are several typhoons that are very, very close to Lüdao, but you can see that the scale of disturbance are generally okay. And I assume that this may be because this island has been suffered from a recurrent typhoon impact, so the ecosystem there is more resilient to this disturbance. Okay, so finally I would like to talk about the anthropogenic degradation. Okay. So we have one long-term recording site of Chaojing, which is actually currently managed by Zhang's institute. Yeah, and this area is quite famous in northern Taiwan because although it's a marine protected area, but because of the rich biodiversity there, so during the holidays or summer diving season, there's an influx of recreational divers. Yeah. So we start our long-term monitoring since the middle of 2020, and you know that 2020 that's the year when global pandemic occurs, right? And in Taiwan, we are still kind of okay, but the problem is that no one knows where we can go, so everyone goes diving. Yeah, and then that's the problem happens. That you can see that in the daytime during the Dragon Boat Festival, that's one of the busiest national holiday in Taiwan. You can see, and I remember that weather in that period is very, very good. So since very early morning, then the low frequency range is dominated by diving noise. That's not the shipping noise because usually ship just passing by the area. So usually it's kind of transient signal, only lasts for 10 or 15 minutes. But for this kind of extended noise exposure, it's caused by diving noise. Yeah. But if you compare to 2021, that's the same holiday period, but in that case, Taiwan is in lockdown situation, so no one can do recreational diving. Yeah. And you can see that the daytime is very, very quiet. There's a dramatic change in terms of soundscape. Yeah. So we compare our data during COVID lockdown period without any recreational diving, or a weekday or day off after the lockdown. We found there's extended noise much longer noise exposure during the day off. So that means the impact of recreational diving on the coral ecosystem soundscape. Yeah. So we are very lucky that because at that time our director still allows our team to do a survey in Chaojing. So we established 4 or 5 recording sites in this area. And if we compare the lockdown period and the weekday and day off after the lockdown, we found that the low frequency range during the lockdown period, it shows some kind of a higher intensity level in shallow water, but lower intensity level in deep water, except this one is very close to shipping lane. But if you look at the data in after lockdown, not only the noise level significantly increased, but also the spatial pattern dramatically altered. Yeah. So this shows that diving activity will definitely make an impact on water soundscape, especially their spatial gradient. Yeah. So think about that you are fish and using this frequency range to communicate. Then the diving noise just a kind of mask all of these signals, and then you will not be able to efficiently talk to your friends, and then even cannot find locations. So this may be an impact for ecosystem resilience. Yeah. Okay, so if you are interested in this data set, please visit the poster number 20, and Jian will happy to share her experience with you. Okay, so finally, I would like to talk about some project we are doing. Recently, we are very enjoyed about kayaking in coral ecosystem because from the previous result, we mostly share result from our long-term recording station, but it can only show one specific habitat.
but cannot really show a spatial gradient. But recently, we have tried to use this kind of kayaking survey to expand our spatial coverage, and then you can see that the result very closely relate to the reef the distribution and the structure. Yeah. And right now we are also using both surveys to do large-scale soundscape mapping so that we can reveal the potential reef habitats. Yeah. So if you do this kind of stuff, combine long-term reporting side and spatial mapping, then you will be able to model or visualize the soundscape dynamics, like a coral ecosystem. And in this case, the Grayson Island, we can even visualize the dynamics of shearwater hydrothermal vent and explore the interactions. Okay, so finally, I would like to say that I hope through this presentation, you will kind of get a sense that, I think soundscape is a missing dimension in our ecosystem conservation. We can consider sound diversity as one of the management topics because if you protect soundscape diversity, then you kind of just protect the behavioral and the cultural process behind this diversity, right? And with long-term systematic monitoring of underwater soundscape, we can track habitat degradation in response to different kinds of stressors. And the future management would be benefit would benefit ecosystem resilience if we're trying to prevent the human activities disrupt the spatial gradient of underwater soundscapes. Yeah. Okay. So finally, I would like to thank all the collaborators. Another I don't know how to dive, so basically all these works have been currently helped by all my collaborators and all my lab members. Yeah. Thank you very much.
We have to wait for the people from another rooms, so we got a few more times for questions. Anyone? Please, Junkie. Did you use the mic? Yeah, just in front of you, on the desk. Yep. Hello, yes.
It's interesting. I don't know if I missed when you were explaining. So you did present some of the results where you say 2020, 21, 22, there was a weakening of the sound because of the effect of heat stress. Did I understand correctly? So it's interesting. I want to know whose sound are you missing or whose sound are you listening in that time? Who are the people who are making sound underwater?
Yeah. So in this case, the sound pressure level is mostly from the crustaceans. Yeah. So because crustaceans cetaceans.
Okay.
Because we know that this signal is very typical in coral ecosystem or reefs, so we consider this as one of the indicators. But we still don't know whether it really represents the ecosystem health or not. But from this kind of time series, we can actually see the recovery after heat impact.
Yes, sorry, because the next slide, although the DHW was very high in 2000,
yeah.
So you missed, right? The soundscape is almost similar. So I missed that explanation. Did you say why it is?
Yeah, so we didn't detect the same pattern in Xiaoliuqiu.
Right.
But we know that in 2022, because we don't have data in 2020.
Okay.
2020 that's the most severe bleaching year.
Okay.
Right. But in 2022, although the DHW showed this kind of data, but actually I think field survey didn't really reveal the extensive mass bleaching, in that year. So that is probably we think, okay, why we didn't detect a substantial degradation in soundscape in 2022? Probably because the inconsistent pattern between DHW and the field condition.
Right. Because the reason I ask you is that I want to know that this kind of decrease in the sound behavior during certain time of the heat stress or when we see the increase in temperature, the animals or the organisms who make sound, are they relevant for us to really address the question of how corals and coral reefs are responding to that year's temperature stress?
Yeah.
So I don't know, I'm just asking you that.
Yeah, that's a good question. I don't know either. Yeah. So I think there's currently a gap between the behavior of these animals and the seawater temperature. We really need some kind of control experiment to understand why this kind of heat stress may impact soundscape. I think mostly this impact animal behavior, this some reducing behaviors, but why this still remains an unknown question to me.
Thank you. Thank you.
And I just want to make a point that most of previous studies on this part mostly focus on the temperate region. Yeah, especially when we talk about the relationship between seawater temperature and this animal, like their sounding behavior. But very few studies have been conducted in tropical region. So that is something I feel very interesting. Although I didn't know really what's the mechanism behind, but this shows some kind of potential direction that we may be able to explore.
Yes, Tina.
Jeff, can you, maybe I don't understand, because you have two sampling stations in Sanfu and Lobster Cave. But then how did you map the entire?
Yeah, okay. Yeah, that is okay, it's my fault because yeah, we have okay, so it's like this, that we do seasonal onboard surveys. So we start by lots of place to collect snapshot recording, but the snapshot recording is not very reliable. So we combine the seasonal mapping with the long-term recording data into our geospatial model, so that we can kind of capture the snapshot between different seasons, and then use our long-term recorder to calculate their time series pattern. So that's why we can predict the spatial change of underwater soundscape in different conditions. And well, we think that this is our first trial to visualize this kind of soundscape trend, but we think this may be kind of useful for predicting what may be the impact area by different kinds of stressors. And right now, for example, Zhao and I were discussing how we can combine our fine scale for example, like this, right? How we can combine this kind of accounting survey with the basic community data to better understand how soundscape can be used to infer a basic condition.
Anyone? We got time for one more question. Yes.
For the exciting presentations, and I think we are getting a very good combination idea to understanding nature's dynamism and statistics, both and relations. Combination is important between agents and science predicting, so like blood tests and EKG test for human health. So I feel that our future is very nice. Thank you.
Thank you. Yeah. I do hope that this kind of not very lots of people are working on soundscape in Coral Reef Society, but we do feel this is quite important dimension for coral ecosystem conservation. So if anyone is interested in this attack, we would love to offer our technique or even resource to establish the capacity in your project. Yeah.
Okay, thanks Tina for this very good talks. Thank you. And the next speaker will be different because he's not from the academic, he's more from the industry. So it's Guo Zhen from the Delta Electronic Foundation talking about their coral restoration scheme.
Hello everyone, my name is Guo Zhen. I am not the CEO of the Delta Electronics Foundation. Unfortunately, Mr. Win Chang had a salmonella infection, so he's in no condition to do this presentation with us today. So I'll be reading off his script in this place. Yeah. My apologies for not being able to replicate his personal charisma. Yeah, I do want to give you a heads up because we are not researchers. But I do hope that this presentation can give you a little bit more understanding in what we're doing as a private sector and what's our approach to collaboration with scientists. So here we go. Yeah. Dear Coral Reef experts, I'm honored to represent the Delta Electronics Foundation in presenting our learning journey in coral restoration over the next 30 minutes. We hope that through our exchange with you, we can encourage Taiwanese enterprises to invest more resources in preventing the loss of their biodiversity, especially in the face of such a comprehensive threat posed by climate change. Delta Electronics Foundation was founded by Dr. Bruce Zhen, who founded Delta in 1971, a global provider of power and thermal management solutions with a worldwide sales revenue of $12 billion US. Delta's business category include power electronics, mobility, automation, and infrastructure. And the foundation tried to use Delta's technology and volunteers' effort to deal with the impact of climate change. The story behind Delta's commitment to corals started in 2019 when IPCC published its special report on the ocean and cryosphere in a changing climate, or SRCC in short. We started to realize an urgent need to enhance climate adaptation in the ocean. 1 year later, when Taiwan experienced the most severe mass coral bleaching, a Delta's engineer, who is also a bike lover, sent us this photo on top left corner of coral bleaching, and we determined to take actions for oceans doing coral restoration. The past 400 days already showed record-breaking sea surface temperature, and this year may even be hotter. Coral reefs are one of the first ecosystems impacted by climate change, and what happened in 2020 showed that actions are needed to prepare for a warmer future. We realized that coral restoration can be an effective way to contribute to ocean adaptation, which was highlighted in the IPCC AR6 Climate Change Assessment Report. However, the report also pointed out that the effect of restoration work will decrease if global warming exceeds 2 degrees Celsius. While the trajectory is currently heading towards a 3.2 degree warming at the end of this century, this means we have a two-front battle ahead of us, mitigating greenhouse gas emission while enhancing the resilience of ecosystems. So how can we do this right? The first step was setting our goals for the coral restoration project. Based on NOAA's guideline, there are 5 main types of goals for coral restoration projects, and different goals correspond to different sets of metrics for outcome evaluation. The Delta Electronics Foundation has prioritized climate mitigation and adaptation works in decades. We decided to keep this core mission and thus take the first one, climate change adaptation, as our objective to do coral restoration. This means our work primarily focuses on preparing coral species well for the inevitable climate change scenario. However, we also identified ecological restoration as an important goal. After all, the main spirit of coral restoration is to create a healthy, self-sustaining biodiversity ecosystem beyond coral species. This would also align better with the 2030 targets of the coming Montreal Global Biodiversity Framework and the Global Nature Positive Agenda. Once we have our goals, we can then start looking at the methods. According to the guidance published by ICRI and UNEP, there are 5 coral restoration methods. Among these 5 methods, we believe coral gardening, which involves transplantation with an intermediate nursery phase, and larval propagation would best align with the sub-objectives under our goals according to the NOAA guideline. These include, the first one is coral population enhancement under the ecological goal. We can contribute to this by planting genetically diverse and healthy coral. As to climate change adaptation goal, we try to reach it through improving coral resilience by selecting and breeding resilient corals, assisting evolution, which means moving corals to locations beyond their historical distribution. In the northeast coast, there has been observations of increased presence of tropical coral species, which are typically in south of Taiwan. We believe it is important to prepare these vulnerable corals in the northeast for a warmer future. Stress hardening at the bottom, which means gradually exposing coral individuals to stress to develop their resilience. According to the UNDP guideline, these two methods on top would have the highest potential to scale up, but come with higher costs. However, we are already seeing research and innovations being done globally to reduce the costs. To achieve the climate adaptation and ecological outcomes we want, Delta has developed a 3-year strategy involving 5 aspects. The establishment of a conservation center and equipment provision. These are the cornerstone of our operations. The facility provides more potential for research collaboration, volunteer engagement, and international cooperation. These aspects together can mobilize the necessary knowledge and resources for the success of our restoration work and the future potential for scaling up. The Chaojing Conservation Center at Badouzi, Keelung, is the base of our operation. It was launched in 2023 in collaboration with the National Museum of Marine Science and Technology, ICOCO, or MMNST. It's a land-based coral nursery capable of cultivating up to 6,000 coral fragments. It can not only provide a sustainable source of coral fragments to be outplanted, but also a controlled environment to research on resilient coral species. An important part that Delta played in establishing the conservation center was equipment and technology provision. A land-based coral nursery can unlock the potential in the sense of both research and restoration methods. But it has been widely acknowledged that the capital cost of a land-based facility is a big challenge for many researchers and practitioner organizations. We realize that there's potential for cross-sectoral collaboration. Delta's industrial automation technology can help build an efficient facility for coral agriculture, as it has been applied successfully to develop the vertical farming system at the Delta plant factory. So we put Delta's technicians together with coral experts at NNST to co-design the coral nursery. For example, Delta's LED lighting solution with programmable logic controller was used to find the most suitable lighting spectrum, which can increase coral growth by 40 to 50% compared to natural lighting. Another notable technology is micro-CT imaging, which can be used to generate 3D X-ray image with pixel size down to 1 micrometer. And with micro-CT, we plan to establish a database for calcium carbonate skeleton for corals. Prioritizing 20 vulnerable local species listed by IUCN, we believe this database can support the conservation and restoration of these species. Currently, the nursery holds around 3,000 coral fragments of at least 11 coral species. Once the coral fragments have grown to 6 to 10 centimeters in length, they will be transplanted to the nearby in-situ coral farm with the help of the Delta volunteers. Delta's coral restoration strategy aims to restore 10,000 corals in Taiwan in 3 years. We also work with eco-angels, Sanhaitans, who are very experienced and skilled in coral restoration to perform underwater fragmentation and outplanting in the Chaojing coral farm. In south of Taiwan, we are also collaborating with National Museum of Marine Biology and Aquarium, TISCR, and NMBA, to restore Hirisian corals in South Bay in Candil. This collaboration aims to cultivate genetically diverse Hirisian corals. We likewise use Delta's high efficiency farming system to facilitate the growth of juvenile corals. Since last year, we have planted 2,000 corals at the South Bay in Candil. On the other hand, we trained our volunteers to assist the scientists with the collection of gametes during coral spawning. Artificial insemination is then carried out in the laboratory. Last year, the collected gametes grew up to corals with a size of 1.5 centimeter from 0.5 centimeter. Artificial insemination really takes time, but it is very essential to a successful coral restoration project. We expect that together with researchers, we can collect 10 coral species and propagate additional 2,000 coral fragments within 3 years. On May 1st this year, while the scientists and the Delta volunteers are collecting coral gametes, we collaborated with NMBA and National Park to live stream the coral spawning event. It was the first coral spawning live stream in 4K quality in Taiwan. The live stream was also commentated by Dr. Chen Dehao and Dr. Wang Lixue from NMBA to also show the public the science behind coral spawning and the research being done to cultivate killer-resilient corals using sexual reproduction. As previously mentioned, Delta volunteers play an important role in our restoration strategy. Although we are looking at the possibilities to integrate more automation technologies into coral restoration, the current method still requires a lot of manual effort to collect coral sperms and eggs. Maintain coral nurseries such as algae, outplant coral fragments, and monitor coral health. We need a group of experienced volunteers who can perform these tasks on a regular basis, as shown in the video. Scientists and volunteers. Every week, the Delta volunteers help the maintenance of coral aquaculture tanks in Chongqing Conservation Center. They also maintain and monitor the transplanted corals in the underwater coral farm regularly. With the help of Dr. Ban Hongyun, 30 volunteers were trained to use the CoralNet 2D coral analysis tool to better track the health of corals at our restoration site. Delta volunteers went through the Coral Watch training at MMST, thanks to Dr. Chen Li-Su, who introduced this tool to us. The tool can provide a more efficient data collection method for evaluating the ecological impact of our restoration work. Earlier this year, the Delta Foundation signed a research collaboration agreement with Mote Marine Laboratory in Florida, together with the 2 museums in Taiwan. Mote is famously known for their coral restoration research in Florida. Up to today, they have restored more than 220,000 corals with a comprehensive approach involving both sexual and asexual reproduction in land-based and underwater nurseries. They are also experts in coral resilience against diseases and heat stress. Through this collaboration, we hope to establish a platform for exchanging research and practical experience between Taiwan and the US. The two museums will use this channel as well to advance their research surrounding the Brazilian coral. Although the coral restoration circumstances and priorities can be very different in different oceans, it would certainly be exciting to see how we can identify the common challenges and spark new ideas together. Later this year, we also plan to arrange some Delta volunteers to be trained at MOLT. I believe most of you already know the fourth global bleaching event is happening now. The distress has been particularly unprecedented for corals in Florida and the Caribbean Ocean. MOLT has shown impressive capability in response to bleaching events, such as the research on post-bleaching diseases and relocating corals to prevent mortality of vulnerable species. Another wave of severe heat stress is expected this summer in both Taiwan and Florida, according to the NOAA's forecast. We need to establish a comprehensive mechanism to respond to potential heat stress so we can plan ahead and know exactly when to do what. We hope to bring back the practical experience through this volunteer exchange. The Delta Foundation has been an observer of UNFCCC for over 15 years and is becoming an observer of UN Convention on Biological Diversity this year. Besides following the latest development in key topics and communicating them with stakeholders in Taiwan, we also collaborate with global organizations on side events, such as 2022, we invited Dr. Chen Hao from MBA and Margaret Lennon, who is the director of the Scripps Institution of Oceanography at UCSD, to join our side event at UNFCCC COP27, where we discuss our approach to coral restoration through technological integration and the importance of marine observation. One of the Delta's advanced technology is 8K resolution projection over 35,000 lumens. Delta Foundation used this medium for ocean biodiversity education and public awareness raising. We believe good communication of environmental issues is critical for building the capacity and momentum in our society for making positive change. This year, we are documenting a coral restoration and bleaching research effort around the world, so people can see the scientists and communities working tirelessly underwater to save coral ecosystems. One of the filming locations is in the Great Barrier Reef in Australia, where corals are seriously impacted by the fourth global bleaching event. We brought together Dr. Guo Zhaoyang and his former mentor at James Cook University, Professor Andrew Baird, who has witnessed closely multiple bleaching events in the GBR region. This is a great demonstration of how corals can connect us beyond borders. To wrap up, I want to share some key lessons that can be applied more broadly in Taiwan. First, I want to emphasize on the need to go beyond short-term restoration projects. Many projects focus on short-term success, which may not stay the same after 5 years, for example. This is mostly due to budget allocation of donor organizations or government grants, which I believe many researchers here have experience with. It is also the same for us. Delta's plan is to restore 10,000 corals in 3 years, and we do need those short-term results, such as the number of fragments planted and survival rate, to inform the decision-making process. However, we need to think about what we can contribute beyond this. This is why impact-driven planning is important. Our main goal is climate change adaptation, so we not only need the corals we plan to survive, but they need to also be genetically diverse and can adapt to the future climate change scenario. For ecological impacts, we will need to find out how the restored corals interact with the ecosystem. such as measuring the size and rugosity of corals and monitoring reef fish and invertebrate communities. A lot of data is required for these metrics, and it is certainly difficult to measure them by ourselves. Therefore, we always try to cultivate long-term, two-way relationships with scientists. Our strategy is informed by the latest research, so we can position our resources and technology better to advance coral restoration research and action. Lastly, an important prerequisite for developing an impact-driven strategy is alignment with global agenda. From the previous experience of the Paris Agreement, companies learned that aligning with high-level global agenda can actually benefit themselves by reducing climate-related risks. In this case, the Coming Montreal Global Biodiversity Framework, created in 2022 under the UNCPD, offers companies and governments a direction to make long-term commitments such as net nature positive targets. Okay, thank you for listening. Thank you for listening.
For the talk, anyone have questions before we head out for?
You can still ask questions. I can try to answer them as much as possible.
I'm really such an outsider. Where did you get the did you say like 600 fragments of corals that you claimed? Where did you get them?
600 in the...
Or is it 6,000 or 1,000? I don't know. I forgot the number, but where did you get those?
You mean those already planted or the capacity?
Yeah, the ones that you planted. Where did you get the corals to be planted?
Oh, where we get the corals to be planted?
Yeah.
Oh, yeah, the corals are selected by the researchers and MMSD from the ocean, and then they are cultivated in the Chongqing Coral Farm, the Chongqing Conservation Center, and then once they reach the stable life stage, they are outplanted into the ocean.
The corals not been sold mostly. Those corals outplanted from the MMSD was originally from the northeast of Taiwan. Yeah, and we got those nubbins somehow because of the dredging of the harbor, and we don't want them to die, so we put them back to the museum and just culture them. Yeah. Yes. Miss.
So I just
Dr.
some naive question. So one question is about, this is my first time hearing this project. So the first question would be, how long you gonna plan to, how long you commit to support such kind of program? It's like just like 3 years program and that's done. And also, are you going to evaluate, like how are you going to evaluate your efforts so far since not? And how are you going to evaluate the protection of biodiversity, recovery, or restoration of the biodiversity?
Yeah, the 3-year target is kind of a long-term strategy, and of course, to achieve this target, we need to develop multiple projects with multiple organizations or researchers. And for evaluation, we are also looking at, of course, like individual projects have their own evaluation tool. But we are also trying to develop like an overarching tools for evaluation according to the international guidelines. So we have like a standardized approach to measure our restoration.
If I understand, so that you mean that you do have a long-term commitment to this restoration program?
Yeah, the 3 years strategy.
Okay.
Yes.
Thank you for the great presentation. I'm just curious if when you do the coral restoration, does fish stock assessment also in the research before you do coral restoration? Do you do fish stock assessment before you do the coral restoration? Yeah. Fish stock assessment. Fish, sorry. Fish stock.
Sorry. That's not part of our, I think that's one of our research projects, but we're not doing it for all of our research.
Anyone have some more questions? No. Okay, then thanks for this talk. Thanks everyone, and Professor Wen has something to talk to you guys.
Thank you, CY. I'm the co-organizer of this symposium in Holland. And it's been a long day. I'd like everyone to join me, give the audience and the speaker a round of applause. Thank you. For people who reduced the dinner with some announcement from the symposium, 5 o'clock, please everyone who reduce the dinner be together at the place we have the lunch box. So there's a space, so get together at 5 o'clock. And for people who doesn't reduce have dinner, in Donghai we have the Donghai Night Market. It's a walkable distance, so on the map you can Google Donghai Night Market. It's a nice place. And we plan to have dinner at a place called Li Zi Cafe. English will be Pier Cafe. We'll try to walk down there and enjoy the landscape of our beautiful campus. And for anyone need a lift from our